Good morning, everybody. Good afternoon or good evening, wherever you're joining us from. We are in the waters, deep waters of Trinidad and Tobago on a research cruise that is, as those of you who have joined us, followed us for days now, is unique in that it is the first research cruise in the region that is led by Trinidadian chief scientist, Dr. Diva Amon, and the majority of the scientists on board are also Trinidadian or from Tobago. And so this is a very exciting opportunity. This is the first time that this is obviously occurring. And today, if you have been following us, we have done a number of dives in soft sediment environments, and then we did a number of dives in methane seeps, which were very exciting and very different than what we have seen before. Now, the methane seeps that we dove on the last two dives were known. They had been sampled before in expeditions that were not led by Caribbean scientists, but Dr. Diva Amon published research on those seeps, and she had participated on one of those expeditions. And so today, we were revisiting sites that we knew had seeps in the past. Today, we have moved a little bit to the southeast from those sites, and we're exploring a new site. We were mapping overnight in search of other areas that may have seepage. And so we think we identified an area where we could see what we interpreted as bubbles, and those would be methane bubbles coming off the seabed, which would be indicative of the presence of methane seeps. But to start off, we always dive in a deeper part of the region in areas that are soft sedimented and they are beyond the influence of cold seeps. And then we move up the slope towards the areas where we expect to find the methane seeps. And I apologize for those using those interchangeably. The more appropriate term is methane seeps, but we often use cold seeps because the temperatures are lower compared to the temperature in the surrounding seawater when you compare it to a hydrothermal vent, which has hot water seeping out. But methane seeps is the appropriate term. So while we're here on the sedimented part, we will do two things. One is we will measure heat flow, and I will let my colleague, Dr. Rachel Lauer, who's an expert on this, talk to you about that when she comes to sample. But before that, we will start by taking a number of sediment cores. And those cores will be used for different measurements. And the different measurements will be animals. So we will count, scientists will count the animals that live in the sediment. And we expect those animals to be only found in the top few centimeters of the sediment, because below that the sediment becomes anoxic and conditions are not very conducive for animals to live in. But there's also some sort of vertical differences. So the very, very top of the core, so the top centimeter of the sediment, may have different animals living in it than the next 2 to 5 centimeters. And so cores will be taken. You'll see it'll be an intact cylinder of sediment that then get sliced, and then each of those slices, 0 to 1 centimeters, 2 to 5 centimeters, is analyzed separately. And so we can see whether there is this pattern of different animals in different parts of the sediment. There will also be analysis that is done for organic and inorganic pollutants, as well as microplastics. And so in order to be able to do all this analysis, we require different numbers of cores. And so we will collect 12 cores today. And so what we will use is, for each one of these analyses, we will use 3 cores. And the reason we do that is because we're trying to get an idea of how similar whatever pattern we find in one core is across the region. If you just take one core, you do not know whether what you see is just isolated to that particular location. So what you do is we take a number of cores, and then you take average numbers of animals or average numbers of microplastic concentrations or pollutant concentration. And that gives you a more accurate representation of what is the pattern in the sediment. So trying to organize a bit here. The pilots are trying to organize in terms of all the different probes and that they need to relocate so they can start removing the core. So typically the way we do this, or again, we is the royal way, the way the pilots do this, is they first take all the cores out and they place them in the sediment, and then retrieve them one by one. So that's what you'll be seeing. As you can see, there's some pretty tricky operations, manipulations that need to be done because the porch is not exactly spacious. I mean, it's very well organized and planned, but it is quite a constricted space, and we have all sorts of equipment on that porch, which is that platform that you see in front of you here, and so you can see the cores are in front of us. Then behind you can see the corner of a biobox, and that is where we'll be collecting our samples. So there's 3 bioboxes of different sizes. Then we have a scoop, and then there are temperature probes, and so it's a lot to pack. It's very sort of efficiently packed, but then also things need to get reorganized as we need to perform different tasks. Okay, we're ready to go. So although we see those bubbles, we see them when we map with multi-beam. So we see them reflecting off the seabed when we're mapping. We haven't actually see them when we are on the seabed. And there could be many different reasons for that. The current has been very strong. The bubbles could be quite small. We've been looking, but we haven't been able to see them. But the fact that we've seen them on the multi-beam led us to the right places. So obviously, they are coming off the sediment, and it was the same pattern that we saw last night for this particular site today. So hopefully, we will find seeps today. Today is more of an exploration dive. The previous two, because we already knew the location of the seeps, it was more reaffirming that the seeps are still active because they may stop being active, and also deploying an experiment, which we can only do once we know what the landscape looks like. So we know their seeps, we know where they are, and then we can deploy experiments that ask questions about different processes, ecological processes that take place in those seeps. And then we collected several animals for analysis, again, for genetics, microplastics, pollutants, etc. So it was in part confirmatory dives, confirming that the seeps are still there and very active, and indeed they were very active, with the spectacular, actually. And also for animal collections. Today, it's a little bit different because we are exploring. So we will be going up the wall, hoping to find seeps. We've seen the signature of them in the water. Oh, bud. Long time no see. How are you doing today?
Good. What's that?
Oh, we're coring.
Alright.
That's why we're all clear,
Any
and
particular order?
just
Doing
take them
one,
out as you can, and then we record them when you bring them back in, when they go. So it doesn't matter, we'll take a picture, we'll
Put
know
them
how
all
you
in
put them.
first?
So just put them, yeah. Not too close, maybe 7 to 8 centimeters apart, but all in front of us.
Yeah, we're gonna. Yeah.
So there is a skill in placing the core into the sediment, because it has to sort of go in vertically, because we're trying to capture an intact chunk of sediment that is the size of that core that you see now. And so it has to go in a sort of smooth motion vertically, and then typically we leave about maybe 5 centimeters, 2 to 3 inches on the top that is full of water, to make sure that that surface of the sediment does not get disturbed by pushing the core too far and having that plug touch the core. So there you go, very smooth movement, continuous movement, and it goes. Of course, the sediment also has to cooperate. Sometimes it's too compacted, it's too clay, more like clay, and it's not as simple as doing this, but
So, do you need a picture of it when we,
up.
with all, like in the.
Yeah, so when they're all in place, we'll take a picture so we know. Oh, that's too deep. That's good. Stop. We need a bit. We need about this much, couple of inches of water between the sediment and the plug. So, so in the end, when they're all in place, we'll take a picture so we know where they were relative to one another. And then when we bring them up, before we put them in the sleeve, we'll also do that.
Why you guys take all the samples at the same place, like one push course?
Ah.
Need a bigger volume?
No, it's because we're using multiple cores to measure the same thing on different parts of the sediment, and that allows us to get a more representative, picture of the whatever it is, the animals or the microplastics in the sediment. If you just take one, and you find no microplastics in it, you do not know whether it's because in that particular spot, it's not a very large area, there's no microplastics. So you take a bunch, and then you take the average. So that's why we do it, to just be more representative. And then, but then you also don't want to have them, kilometers apart. And there's also, we're looking at, or scientists are looking at, how many microplastics are in the area where those animals are. So they're trying to find relationships between the amount of microplastics or pollutants and the types of animals that are there. So
this time I'm going to help you too, so you have to watch too. To put one of your cameras here for you? Yeah. This one. Yeah.
What do you need? Okay. The current seems a little bit weaker today, quite a bit weaker today. You can see the current, because we're sitting, we're not moving. You can see how fast the particles are moving past us and the distance. In the last couple of days, we had very strong currents, and it was a little bit of a challenge, both to sort of sample for and for the ROV to move against the current, because we put a transect for them, and then that transect sometimes the movement can be against the current. Perfect. Yeah, looks good. Perfect. And so it always makes it a challenge, but today it seems like it's a little bit calmer down here. Okay, perfect. That's good. Yeah, that's a good 2 centimeters. Perfect. And often what we sort of, the audience admires in the pilots is their skill in terms of the manipulations that they do on the seabed, but there's also another skill, which is, obviously the piloting, the moving of the ROV. And sometimes it's relatively simple for them, but sometimes becomes complicated. For example, the other day we had currents at the surface running in different direction than the currents at the bottom, and so the strength of the current changed as we were going down. But we also have a very long umbilical cord behind us that weighs a lot, and has a lot of drag because it's over a kilometer and a half above us, and so that also is something that the pilots have to take into account, how the ship is moving, etc. So it's not simple, oh, just drive the ROV. There's a lot of different calculations and factors that they need to take into consideration when they move. And then maybe some wouldn't,
I'm just gonna write it here.
yeah. Hmm. Looks good. Great. Perfect. Yep.
Sure that point. Thank you, jamming up yourself. See where you jamming up yourself.
That may be too, yeah, maybe a little forward, because that may be too close to the other ones.
Yep.
Yeah, that's, looks good. Yeah, that there looks great. Perfect. That's great. That's good. No more. Perfect. Thank you. So what you see going by all those particles are whenever, if you see really close, then you'll see that that's sediment resuspended. But when you see in the distance, you see larger particles, and that is what we call marine snow. And those of you who live in the north, you will know, or in the very south, you will know why it is called marine snow. But basically, it's just dead stuff, dead plankton, dead dandruff or organisms that live up near the surface, pieces of food that if they're sloppy feeders, and so all that material rains down, and the term is called marine snow. And so in environments like the one we're sitting at right here looking at, that is the main source of food for the animals that live on the sediment and in the sediment. There is no sunlight down here, so there's no photosynthesis, there are no plants, and so all they have access to is this marine snow and also the occasional great, thank you, the occasional piece of seaweed, and we've seen that there's a lot of sargassum actually that we see often, especially you can see some on the seabed here, and the occasional food fall. So those are big pieces of either wood or plant material or more often animals. And we saw, I think it was the third night, or all the dives are blending now, a ray. And so animals, of course, they will die. And big fish, sharks, whales, mammals, all the animals that swim at the surface at some point will die because of old age, disease, or whatever, and they will drop straight into the seabed. And so again, that is also a source of food, but it's a more ephemeral source of food. And so the difference between the environments that we see here, which rely on this source of food that rains from above, between these and the methane seeps that we'll hopefully find later and that we have seen yesterday and the day before, is that organic carbon is produced locally. And the way they can do this is because of the methane seepage. Microbes, bacteria can use that seepage and oxygen to essentially perform the similar process that plants do at the surface, at the sea surface on our land. So instead of photosynthesis, it's called chemosynthesis, because the energy to carry out that reaction is what fuels the whole process of carbon production. And so, because that carbon is produced locally and it produced in large amounts, you can have this sort of microbes and bacteria that replace the plants. And so, then those have large populations, and those bacteria can then support a much richer ecosystem in terms of biomass. So that's why we can see all those mussels, and we saw tube worms and other types of worms because of this local source of food. And so chemosynthetic environments such as methane seeps and hydrothermal vents are considered oases in the deep sea, which is generally energy depauperate because there's no other primary production. And so an interesting sort of line of research that has developed in the last 5 to 10 years is how much do those seep ecosystems support the surrounding environment. And so the idea is that there is some transition between this high energy, locally produced high energy ecosystems, that such as the ones that we see at the cold seeps, and they're unique to the cold seeps. Those animals don't survive away from the cold seeps, but they also die and they release dead, material, organic material in the surrounding waters. And so that that material can then be richer than just simply what is raining from above. And so we get perhaps what we call animals that live in, we call the regular deep sea. So that's the deep sea that is not impacted from directly from the cold seeps or doesn't rely on the seepage. So those animals start getting closer and closer to take advantage of that increased organic production at the seep. And so we're trying to also understand what is that transition. So how do we transition from the very dense, high biomass ecosystems that we see at the seeps to the types of ecosystems that you see on your screen right now?
You can grab it all over like this. If I choose it this way. As I lift the whole thing. Stop. It's just moving, but just tight. The sleeve.
Mhm. we can start putting them on this side.
If you need to go on that side, you need to come to the right first, and our porches, because you're already touching it. Yeah, I don't think. Go to the right.
A little bit to the left. Yeah, that looks good.
And you could bring this one to the right if you want. More.
Great. Yep, that's good. Oop, nope, not that deep. Don't want the sediment to touch the top of the tube. That's good. Perfect. Now, I think that anthomasters that you see there has to be on a rock because they don't grow in the sand. So that whole area between 4 and 7, and you see the little anemones there on the side, that maybe have a rock underneath, so you may not want to put a core there. You may hit a rock, so avoid that area and maybe come this way or to like 7 o'clock from number 7, something like that.
To the right.
Yep, perfect. That's good. Let's see. Yeah. So we're almost done. We have done 9 out of our 12 cores. Again, we want to take all those cores and relative close to one another, so that we get a representative area, a representative idea of this particular area. We take multiple cores in a particular area, and then the cores are taken for different measures, and so we need the cores that are taken for the animals to be close to the cores that are taken for the microplastics, so that we're able to draw relationships between where the plastics are and where the animals are. It'll be too close. We need it either over on that side or closer to the sub. Yeah. Can you go? You can't go any further. Yeah. Yeah, that looks good. So we don't want them to be too close, because we don't want pushing the core in to sort of affect the sediment next to it, to the core next to it. And so that's why you see we keep them at least 60, 70 centimeters apart. But we also don't want them to be meters apart, because then that's not similar conditions. That's good. That's good.
You have to go this way, right?
If you want to bring it
Reach isn't very good over there. Why can't I put it here next to 4?
next to 4.
No, I just told you to go this side so I could watch in, because with the edge of the
I'm worried about the rock there. You may want to come more to the left.
To the left.
Yeah. So there's that rock there. I'm not sure how big. Yeah, there.
You need to come up with this. So you need to come up with your elbow. Yeah.
And I can just shake the segment out of there. Can you? Yeah, just perfect.
Just shoulder down now and elbow down. Yeah, that's better.
Great. Good. One more, right? So here's our last one going in. So we don't want it by the rock, and we don't want it in that area that you've already tapped on it. So we don't want it here either. So if you could put it there. It's not. Yeah, it's good. Okay, left side of it. Great, it's perfect. So maybe if we can zoom back a bit and take a picture of the second. And maybe we can have the lasers on so that we can get scale, so we can measure distance. That will be wonderful. And maybe not the arm in the way. a nice clear view. That's good. Are the lasers on? I can't see them. Why can't I see them? No. Ah, see them, yes. Right. Perfect. So if we could take a couple of shots of this. Perfect. Thank you. So that's all for me today. I will pass you on to my trusted geophysicist colleague, Dr. Alicia Lauer, who I know has a lot of fans out there. And she's going to be overseeing the collection of those cores and then more heat flow measurements. So thank you all for listening. Over to Rachel. Bless you. Yep. Okay. Yep. How's it going? Good. What's that? This is a bit weird, but all right. So 12 made it. The cursed.
They're not back in yet.
All right, we just want to put them back and make sure we get a shot of it in front before it goes in. I think that's it.
Does it matter which order we were coupling?
I do not think it matters. I'm just going to write them down on here anyway, so it's up to you. Whatever order your heart desires. I think they should be.
I'm not sure.
Uh-oh.
But you should go for 5 because if I porch out, I think 5 is the limit. We'll be able to porch more than that. Okay.
So they might be a little different lengths. The push first?
Yeah, I mean the all the same length is just the stop.
Oh, okay.
Some are short. See like 3 is short.
Oh, yeah, the top thing you grab. Yep.
5 is short.
Right. 6.
Should we put these in the difficult ones out of the way first, or just work from port to start?
The only reason why I told you get 5 I don't like the port give you the whole porch out.
Yeah, I
Like
mean,
it.
where are we putting it in the porch?
Yeah.
Should we work port to start with?
Yeah. Because
So
the
they can
corner
go-
ones, you'll be able to like show them how better.
All right, so good afternoon. This is Rachel Lauer from University of Calgary. And we are picking up the 12 push cores that were just collected. These get recovered, brought back to a wet lab, and on this cruise, we're mostly looking at macrofauna, So little critters that are living in the upper part of the sediments. We actually sieve and separate those fauna from the sediments and look at those under a microscope. Some of the cores will also be split and frozen for other analyses. It really depends who the scientists are on board in terms of how those sediments are used. And sometimes they're used for estimating sedimentation rates, so how quickly the sediment is piling up on the seafloor. There are tiny little organisms that are floating in the water column, and when they die, they settle and become these sediments. And there is an entire area of geoscience that uses these tiny organisms to date the specific time that it was deposited. They're called foraminifera, or forams for short. And they can also even tell us something about like past climates.
Which one you want to go?
Get last night.
Start off with what you want to do.
I think another one of the analyses that we're conducting on, well, not we, but one of the researchers on the ship is looking at organic pollutants, so trace metals, maybe stable isotopes, to look at conditions in the past, so when these sediments were deposited. So we get the top, I think, 40 centimeters. And depending on the sedimentation rate, that can be thousands of years ago when we had different conditions present in our oceans and on the planet. Is that number 2?
Yes.
Thanks. Alright.
Let it go now. You have to like post it in that graph. Just touch it on.
So last night we spent about 10 hours mapping this area, which has not been mapped previously, and identified a ton of additional structures that are probably similar to what we dove on yesterday and the previous day. And these are active seep sites that are just, at least yesterday's site was covered in mussels and other chemosynthetic animals that thrive in this methane-rich environment. And we got to see some of the structures that get created by just thousands and millions of years of active seep communities that are thriving and then subsequently dying and providing a platform for the next generation and the next generation. So just crazy structures at the seafloor. And we saw methane hydrate, which is kind of a wild thing to see. It's clathrate is an ice cage that forms from water molecules that are surrounding the methane. And it's stable at these pressures and temperatures. If you bring it back to the surface, it is very dangerous. But at the seafloor, it's a byproduct of all of the hydrocarbon that's present in the subsurface. And cool. 11.
push down. I don't know. I think it has to push down somehow. If you watch number 2 and number 10, height of it.
Is it spring? Nine. Is there depth here? So if you're just joining us, we are east of where we were yesterday, still on the Barbados accretionary prism. We're in a low spot that is adjacent to what we assume is another one of those carbonate mounds. And first, before we start any transects or do any other measurements, we're collecting 12 push cores in one location. And those get used for all sorts of analyses, both on and off the ship. That was one. You can see these sediments are super sticky and clay rich, so it's not like sands as much as it is really, really fine grained material that's mostly made up of these single-celled organisms that live in the water column and are either silica or carbonate, and their sedimentation is primarily the result of them just making their way back to the seafloor after they die. So the sedimentation rates are not as high as closer to land, where you have rivers and other water bodies that are carrying lots of sediment to the marine deltas, and you get really high sedimentation rates closer to land. But here, it's probably on the order of, I don't know, a high sedimentation rate would be like a centimeter a year, and here it's certainly much less than that. There's an entire branch of geology that's focused on those little critters that can act like a time capsule. So the deeper stuff is, depending on the deposition rate, can be thousands of years ago, when climate was different and there might have been different animals present in our oceans. And the people that specialize in the analysis, those analyses can tell a lot about the conditions that were present at the time those animals were alive, which is really cool. So they spend a lot of time sorting those tiny little animals and analyzing them under microscopes. Transects are just, sorry, I may not be speaking clearly. A transect is just a line that we draw while we're planning our dives. So overnight we map the area, get up and process and analyze the data and the maps, and then pick places where we want to drop in based on what kind of measurements we want to make. And a lot of the mapping work, because this is primarily biology and trying to take a census of who's here, because no one's, you can make a map from a ship or even from space, but really what's important for this cruise is identifying what creatures are present and investigating different biogeographical locations within Trinidad and Tobago's waters. So we started on the shelf, so very close to land and pretty shallow. And then we focused on a site a little bit further, but still in one of the back basins, so fairly flat. And now, since then, we've been focused on these carbonate mounds. And a transect will just set up between two waypoints and try and move at the same pace using the sub. And not stopping to sample or do anything, just identifying the creatures that we're seeing as along that transect. And because we're recording with really high resolution cameras, we can go back, or not we, but real biologists, not me, can go back and analyze those data and identify all of the organisms that were present along a specific transect. Yeah, so some of the things you can see in these cores, if you are one of those geoscientists who's looking at the sediments and getting little snapshots of past records. They're called paleoclimate researchers, and some of the things that they can identify within these cores are, everything from like microfossils, like those foraminifera. They can see records of like algae, even charcoal. There's widespread fires that are providing ash layers. There can be evidence of volcanic eruptions within the sediments that we sample. And then between all of those sort of events and little snapshots of time are just the background sedimentation rate that's happening. And if we know that background sedimentation rate, we can use that to estimate the timing of these other events, like fires and different environmental conditions. Even how much it rained or how much precipitation was experienced in a given time period. And then closer to land, if you do these, you can identify records of earthquakes.
Yeah, these are most corner ones.
And those types of studies generally use longer cores, so piston cores, but it's the same idea. Instead of doing it with the robot, you're just putting it over the side of the ship and allowing it to drop to the seafloor. And then a piston is like literally triggered and shoots into the soft sediment, and then you collect a really long record. And depending on how long those cores are, you can get millions of years ago snapshots of conditions that were happening back then. And it's that kind of analysis that's allowed us to understand how often big earthquakes happen. Because what happens when you have a big earthquake on land or at the bottom of the ocean, there's a lot of ground shaking, right? So what that does is causes very rapid movement of sediment from the continental shelf into the deeper ocean. And that layer that happens as a result of that ground shaking provides a record of that earthquake. And if you know how sediment is deposited and at what rate, you can go back and determine, okay, it looks like we had 2 of those sandy deposits that represent earthquakes, and based on sedimentation rate, we can say, okay, it looks like these happen every 50,000 years or 5,000 years or 500 years. And although that doesn't really allow us to predict earthquakes, it does give us an estimate of what's called their recurrence interval.
right side for now. Let's finish the course.
What you want?
Any other side. Front the, put it in front the ramp. Higher, serial armrest. So I don't know if you could go up with your elbow more. If you come up with your elbow more, might get a little more height. I'm not sure if you can go. Right. It's really good is if you put the collar more horizontal and come in. As soon as you come in, you straighten up the collar. So you go like this. Is it good? It's so right now. Turn it a little more so. Come in. As soon as you're coming into the hole, you straighten up. And then put it in. Watch the far from it so you can straighten up now.
Yep.
It's a good tip.
Yeah.
Yeah.
Is it? It's not going to be disturbed back there.
Got it.
We can't see that. We'll have to turn the camera on.
Get through.
Yeah, the seafloor is sticky. Yeah, the texture of these sediments is like, I mean, here it's almost like peanut butter because it's these clay particles that are really small. And unlike sand that doesn't really have any cohesion or stickiness, these are the opposite of that. They tend to stick to each other. So imagine like driving through mud or something that is clay rich and it's sticking to your tires and sticking to itself, versus driving on a beach where, you know, you're just gonna compress the sand, but it's not actually gonna stick to anything. So these are particles that are, again, just like single-celled organisms that are made up of either carbonate or silica. And because they're so small, they tend to have a lot of cohesion is the word, but stickiness is another word for it. So once we collect these cores, we'll be actually probably stay here to do a heat flow measurement and then head towards the mound.
We need to some the other one is going to block you this high energy. So that's the big
Mm-hmm. Bless you.
a little too critical now. It's a come on a higher, yeah. Yeah, like that angle is good. Shoulder, left hand, the two teachers.
So if you are just arriving, you haven't missed much. We had a bit of a later start today. Spent last night mapping this area, and we're still on the accretionary prism. So lots of deformation and scraping of sediments and piling them up. And we are focused today, once we are done with the core sampling and some heat flow measurements, we're heading towards the mound. This is another carbonate mound. They're all over the place here because this Trinidad and Tobago's waters are basically a hydrocarbon province. So there's a ton of methane and CO2 and hydrogen sulfide that is making its way up from about 10 kilometers below us, where those hydrocarbons are forming, where it's nice and warm and high pressure. And then they migrate upward along these faults that develop as a result of all of that deformation, and eventually make their way to the seafloor. And over millions of years, the chemicals that are associated with those hydrocarbons are interacting with microbes, bacteria, and forming a platform of carbonate that creates a place for all these mussels and shrimp and all the other species that were observed yesterday, and forms these wild carbonate structures that are also combined with the fossils of the animals that were living there. So instead of being like a part of the crust itself, these little mounds, well, not little, I mean, yesterday's mound was about 300 or 400 meters tall in some places. And that's just the result of these chemical and biological interactions that are taking place at the seafloor. And one of the things we're looking for in terms of deciding where to dive is the signature of bubbles in our multi-beam. And those will show up because bubbles have a different density than water. They have obviously air, so much lower density. And we can see that contrast in the multi-beam data. At this depth, however, those bubbles are very small, and as we've seen, they're very hard to find a bubble stream. But we know there's a lot of methane because of the animals that are living on these mounds. And then we've also seen a mound that represents an ancient methane seep. So not active at the moment, but was previously active millions of years ago. So, power to the probe, and then the camera. Yep. Cool.
You can see it down there.
Where is it? Oh, there it is. Okay.
Can we also turn on the temperature? Where's the display?
Oh yeah. Do you have the temperature, like bottom water temperature displayed somewhere?
It's in the live stream, 4.6.
Oh, it's in the live stream. Okay. It's not over there. Is that where you want it?
Yeah.
Okay. Are we able to see that over there? So I think as long as it's not disturbed where the probe is, and it doesn't look disturbed. We don't need to poke, right?
We just poked a bunch of stuff.
Yeah, we just poked
It's
like
fine.
12 times. Okay. All right. So, can we get power to the heat flow probe?
Can you power to that?
Yeah, it's this guy. And then we just keep an eye on it because if there is a rock, it'll break the
Different name.
heat flow probe. So we'll that one. You got power?
Yeah.
There's a what? Yeah, that's fine. Yeah, that could be anywhere. Yeah, we don't know. What?
Oh no, it wasn't supposed to go in yet.
No. Okay, shit. All right. We might have to move. So,
I was just sampling the water bottom.
yeah, we have to get the water bottom temperature first. What do you want to do?
We'll have to
Yeah, we need, okay, so for this, we have to collect bottom water temperature for a few minutes so that we know how temperature is changing under us. So we wait like a minute and then go in, but so we're gonna have to probably move because
not very far.
yeah, we can't go into the same hole because we won't get any friction.
I'll just put it at the bottom.
This is good, yeah. Whoa. All right, so yeah, we'll sit here and just wait for the dust to settle. And
Okay. Calibration's done.
yep, we just need to be able to see it, so. What's
No, you're not doing anything.
Is that from me?
Nah, that's from when I.
Oh, this is just our heat probe. That's,
no, it's
This
the
is from when I jumped. It's just following us.
yeah, because are we moving in the direction of the current?
it's the armor covering this course.
All right, ready?
Yeah.
Okay. Just keep an eye on it. So if it hits a rock, we'll stop it. Ready to put in the heat flow probe. Awesome, thank you. All right, so we are in the process of taking a heat flow measurement. Because this is a new location, we will take both parts of the measurement. The first part is, how are we doing, Rob? Is it vibrating? Okay.
Their names look the same as the last ones.
Yeah. So the first part of that is we use a titanium probe that has a lance that's about 60 centimeters long, and titanium housing with all the electronics that's basically plugged into the sub and allows us to see our data in real time. And the first thing is just pushing that into the sediments. There's 9 thermistors along those 60 centimeters, giving us a sense of how temperature is increasing as the depth increases. And what happens initially is you get frictional heating when you push it into the sediment and allow that to decay so that we can see what the actual temperature is, not from the friction, but temperature below the seafloor. And then after that's stabilized, we fire a heat pulse, and that is a known amount of heat that goes in over the length of the lance. And then how that decays tells us how conductive or how easily heat moves through the sediments. And then between those two measurements, we can get what's called heat flow. And that's how much heat is actually crossing the seafloor. And there's heat crossing the seafloor everywhere because the Earth is cooling, A, and also we know how much heat should be crossing the seafloor in a given location because we know how old the seafloor is. So younger seafloor has higher heat flow, and as it gets older and colder, it's less heat flow.
It's touching first, so you have it like this, so it's touching like this and this.
And if we use this heat flow to do a transect, which we're not going to do today, we're going to get a couple of measurements and then straight to the carbonate mound. But if we do enough of those measurements, we can start to get a sense of where fluids are coming out of the seafloor. And those could be, in this location, those would be faults or other things that are connecting the seafloor all the way down to the plate boundary, which in this area is between 8 and 10 kilometers below us. So this probe is, the pilots here came up with a clever way to mount it onto the side of the sub, and it's basically on a hydraulic ram, because if you want to measure temperature and how it changes with depth, you want it to go in perfectly vertically. And before we started working with Schmidt, we would have it in the arm and then try and push it in straight, which is really hard to do. And now we have it mounted on the side of the sub, and it's just goes in perfectly vertically. We just have to be careful, make sure we don't hit a rock or something, because titanium's strong, but the sub is stronger.
I mean, if you don't have it rope straight, you have it rope not twisted like this. You pull in here, but you have it rope twist like this. You just have to rotate. That's not.
Yeah, this we usually take the pokey thing, also known as Excalibur, to push into. We use the pilots use the arm to kind of poke the sediments and make sure that we have enough sediment. Of course, there could be a rock or something hiding within the sediments, but we want to get a general sense of, do we have enough actual sediment to safely conduct one of these measurements? So I don't think you guys can see it, but we have one of the many cameras on this cool sub is pointed right at our heat flow probe, so we can make sure it's safe. Especially because today we are going to be flying around lots of carbonates and boulders and structures that may cause harm to that instrument.
How to almost
So again, like one of these measurements takes about 15 minutes. As exciting as it is for me, I know it's probably not why most people are here, but don't worry, we're gonna get to the fun stuff later in the dive. As a geophysicist, this is fun for me, so. How does it look, Rub? Same? Yeah. Wow. Okay, cool. Okay, so I'm talking to Rob, who is a postdoc in our group at the University of Calgary. He's a geophysicist, and in addition to doing heat flow, we're also helping with the mapping and again, this part of the seafloor has never been mapped from a ship. So every morning we get an amazing gift of seeing all these new structures that have never really been mapped before, providing us with a bunch of different options as far as where to dive. And we are primarily focused on areas where we see bubbles, not with our eyes, but with the multi-beam. So the multi-beam is using sound to along a swath. So it's mapping directly under the ship, but out to the sides as well. And then that swath is how sound travels from the ship to the seafloor and back through the water column. Gives us a sense of all of the topography or bathymetry of the seafloor. So we can map those mounds, and then we can also see little plumes. We can't pinpoint exactly where they are because of the ship's moving, but we know what area those seeps are occurring, and those are really good indications of where we have scenes that are similar to what we saw yesterday, which was just piles of mussels and tube worms and yeah, all sorts of creatures that are thriving in that methane-rich environment. Heat pulse? Okay, Cool. So we just fired the heat pulse, so we got another 7 minutes of excitement here. So after this, we're gonna get a little bit closer, start a transect towards the mound that last night we saw. I think we found like 4 or 5 places that were bubbly. A lot of these mound structures that are actively streaming methane from the seabed. And we picked this site because it appeared to have, not quantitatively, but qualitatively, the most bubble streams that we saw during our mapping. Yeah, I have never seen that many mussels in my life, although I don't think I've ever... What? No, but okay, so on the west coast of work, Rob and I are from Canada. We're from the University of Calgary, and we have worked in cold seeps on the west coast of Canada. Yeah, but not this many mussels. We didn't see this many. This is insane. Like, just mussels everywhere. And we only saw probably 10% of that mound, which is bonkers. So this is much deeper than those vents. And also different, yeah, I think the depths is also going to influence who's living there, obviously. And it's harder to find bubbles at depth because the pressure is higher, so the bubbles are smaller. Maybe we should go until we find an animal and then plop down to the next one. We didn't plant waypoints or anything. No, I know, but for our measurements, we didn't. Yeah. Okay. Hmm. Hmm. 4 more minutes? Okay. Roughly? Okay. doing 2. So, I mean, if you want to.
No, no. Let's so the chip will come.
Okay. All right, so we are done with that measurement. We can take up the probe, and we're going to head towards our first waypoint. And as we get closer, if there's something cute to look at, we can sit down and do another one.
Oh.
Because give the fans what they want. Yep, this one. We just did one. Yep, we're doing one more and then we're done. I'm trying to look for something charismatic so that they're not looking at the mud.
Oh, no. I mean, you can do a transect.
No, we're not doing a transect. We're just doing 2 measurements. Yep, as one
So
here
then
and
one
one closer to the mound.
Okay, so what I'm saying is we need to do a transect. So,
Yeah,
so
I mean, if you want to,
can we do a transect now and then do a measurement?
I was just going to go like 100 meters.
Oh, okay.
Yeah.
Ready?
Yes. Oh, you weren't talking to me.
Yeah.
Okay. I don't have to do anything. Just-
That's to move straight away, Prof.
So we're just gonna go like 100 meters closer, and then we'll do one more of these, and then we're done with heat flow.
Bridge already.
Are we ready to go ahead?
Hi Tom. So that's us on the graph.
So we did not see any bubbles on any of the recent streams. We know they were there. It was honestly this area where we were diving yesterday and the day before had such strong currents that it would have been tough to spot the bubble streams. Places where I've seen them are much shallower, so the bubbles are obviously bigger. Not obviously, but under less pressure, bubbles are a bit easier to see versus where we are here, which is, what are we at? 1,400 meters water depth. We have lots of people that speak different languages, Portuguese, Spanish. My Spanish is enough to get by, but not good enough to talk in front of other humans who are listening. Okay, we've got a little sea cucumber off to the right. Yeah, so there's lots of different animals that we saw yesterday, and, you know, we saw shrimp, we saw crabs, we saw giant isopods, we saw these guys, sea cucumbers. Oh, is that another? Nope. Little coral on a sargassum there. We saw living within the mussels were lots of these ghost white chemosynthetic shrimp and eel pout, which is my favorite new little fish that hangs out in the mussels. They're like little tiny eels that have like a little blue spot on their head.
Okay, I'll come level with you.
Thanks, Tom.
What else did we see? Octocorals. I think on this, in the water column, we've seen a bunch of siphonophores, which are so cool. And yesterday we got to see methane, or methane hydrate, which is like called a clathrate, which is basically an ice cage that forms around the methane, and it looks like a slushy or something. It's like snow, and it's basically in the place where we were diving yesterday. There's so much methane coming out of the seafloor, and there was like a roof over top of it where all of that methane was building up, and it formed this giant pile of methane hydrate, which was wild. Are we 100 meters from the last spot? Oh, we are. Damn, that was fast. Okay. All right, so we're ready for another one. We're going to do the pokey thing first. So we're going to do another heat flow measurement here. First, we're going to check to make sure the probe has a full 60 meters of soft sediment. 60 meters. That would be a bit unwieldy. Sorry, it's 60 centimeters. Oh, God. So this is Excalibur. Da, 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 da. Okay, so, and what seemed to work yesterday was holding it once it's in, and then like using it as an anchor. I don't know if that helps, but yeah.
There's not too much current, but we can certainly do that if you want.
Well, it's up to you. I mean, it was more of a current thing, I think.
Okay, you ready?
Yep. All right, looks good. Okay. Do you want a couple seconds here? It's already calibrated? Okay.
Anytime you're ready.
So we are ready. We'll just keep an eye on that to make sure we're not hitting a rock that we can't see. Cool. Awesome. It's all the way in. Cool. Thank you. Oh, there you go. All right. Yes, you are correct. There is a pressure and temperature range over which methane hydrate is stable. And one of the concerns of warming oceans and climate change is as our oceans get warmer and bottom water temperatures increase, that also means that what's trapped in the sediments is that temperature is going to increase. So you can get what is currently trapped in the seafloor making its way into the oceans, and depending on ocean chemistry and the depth and a lot of other things, it can make its way into the atmosphere. So that has been the focus of a lot of research, looking at places that are monitoring for bubbles in coastal areas that have a lot of methane hydrate trapped that has now been mobilized by ocean warming. And that obviously, the deeper ocean, it's affected less than shallower parts of the ocean, but it's affecting the whole ocean, of course. It's just that it's felt more in shallower parts of the ocean. How are you guys doing? Good? 2 thumbs up? All right. Good. Yeah. This ship rolling makes me sleepy. Like I want to be in a hammock, but
We have that.
I know there's a hammock. I haven't been in it yet, but okay, so again, for those of you who maybe just showed up, we are doing a heat flow measurement. The first part of that measurement is pushing that 60 centimeter lance into the seabed, waiting for the frictional heating that happens when that it gets inserted to calm down, and then we get a sense of how temperature is increasing or changing as a function of depth. And we have 9 thermistors on that 60 centimeter lance, so we get 9 measurements of temperature. And that gives us a geothermal gradient. And then the second part, the second 7 minutes of this measurement, is estimating thermal conductivity. And to do that, we use a heat pulse. So a known amount of heat goes along the lance, hits each of those thermistors. So there's a huge spike in temperature. And then the way that temperature decays as a function of time tells us, and the shape of that curve allows us to estimate what's called thermal conductivity. And that's how easily heat moves through the different layers of the sediment. And those two together give us heat flow. And heat flow is a tool that is very important for people studying hydrogeology, or how fluids move between the ocean and the ocean crust. In this case, we're on a subduction zone, so we've got the South American plate diving under the Caribbean plate and scraping off all the sediment, and in the process forming lots of faults that connect that deep plate boundary all the way to the seafloor. And in some places, we have fluids seeping out at the seafloor. And this type of measurement can help us isolate where those fluid seeps are located, because they tend to have higher temperatures and higher temperatures than you would expect based on the age of this crust. And I should know the age of this crust, and I don't. So,
That's what we're here to find out.
yeah, exactly. 100 million? It's really old. When geologists say, like, oh, this is young crust, and it's like 5 million years, it's just a matter of scale. So we talk about things in giant ranges of scale, like temporal and spatial scale. So 100 million years is really old. I know, but we're not that kind of geoscientists. We're geophysicists. But yeah, it's really old. And we are, again, on the biggest accretionary prism on the planet, the Barbados accretionary prism. Some subduction zones have almost no prism. This one has a ginormous prism. It's like 10 kilometers deep, and it extends like hundreds of kilometers east to west. So,
Out of my own curiosity, can you explain what that is?
accretionary prism? Yeah. So, in subduction zones, you have one plate that is diving under another plate. This usually happens like at plate boundaries between land and the ocean. Here it's happening to the east, and you have the the South American plate diving under the Caribbean plate. And because of the way the plates interact, all the sediments on the plate that's going down are getting scraped onto the Caribbean plate, and after millions of years of scraping, you just get this pile that is also getting reworked and consolidated and squished together, and then you get faults that form in it. And not all subduction zones have accretionary prisms. Some of them, the plate just goes down, and that's it. And it takes the sediments with it, and there's no accretionary prism. Here, there's a lot of sediments that are coming in, and they are forming this massive structure that we've been on for the past like 4 days. I didn't know what an accretionary prism was until I started my PhD, and I ended up studying them because I was a hydrogeologist and I just thought about fluids. But yeah, this is more like tectonics, like large scale stuff. And then the tectonics here is even more interesting because you have not only one plate boundary between the South American plate and the Caribbean plate, which is a subduction zone, but you also have a strike slip fault. So the same fault that ruptured and caused the earthquake to the west, or a series of earthquakes, is also part of the strands that ruptured are also extending through the top of Trinidad. And that forms the plate boundary between the Caribbean plate and the South American plate, except instead of going under each other, they're sliding past each other. So the geology here is really complicated. When I knew we were coming here and opened up some maps to start studying for this, I was like, uh-oh, I might need to consult with somebody. It's a very complicated environment. How we doing? Same story? Cool.
Do you think the earthquake that recently happened in Venezuela has any effect to
Oh yeah.
the methane here, or maybe what we're looking at?
Oh, that's a good question. What I can say is a lot of times we look, like this was a 10-kilometer deep earthquake, which is really shallow.
Yeah.
So lots of ground motion. It was a strike slip motion, but in the area near the fault, they had surface rupture, which means the energy made it all the way to the surface, and they had like huge offsets at the surface. So like a step formed along the fault between them, like tens of meters of slip, and then, which is unusual. And anecdotally, I have seen articles in Tobago newspapers that even in Trinidad newspapers, Trinidad and Tobago, but in southeastern Trinidad, there were beaches that were like, people go to this beach every day and it's beautiful and whatever, and they come out after the earthquake, and what was underwater is now above water, like 20 feet. Well, it said feet. I'm not sure why feet, but you know what, meters of elevation change. So all these animals that were underwater are now dead because they obviously can't survive out of water, just as one line of evidence. But then people are going out and actually measuring how much uplift happened, and that's way far away from the earthquake.
Yeah.
So yeah, you can have, like in other studies that we've worked on, we like not methane seeps, but like vents, hydrothermal vents that turn on and off after an earthquake because the stress state has changed. Like all the different faults and things, when you have one move, it's clamped down in one spot and then it releases in another spot. So like one event can turn on, but then another one turns off because it changed the stress state on the next one.
Yeah.
So yeah, I'm sure there were things that I'm curious to see if there's any of that elevation changes, but we're making the first map, so we wouldn't know.
Yeah.
But yeah, I'm sure there's offshore changes that were the result of that shallow earthquake.
Yeah. We have worked where we had no maps from a certain area. And
Yeah.
It was a massive earthquake, which turned into a tsunami.
Oh.
In kind of the South Atlantic, basically.
Yeah.
And it's actually really, really hard to prove that you,
Yep.
even if you know something has shifted, it's hard to prove it because it's like there's so much error
Yes.
that's just inherent.
So that is so the one place they did do this.
Yeah.
So you know the giant, well, the Tohoku earthquake in Japan was one of the biggest ever, right? So it was like a 9.0 or 9.1 or something terrifying. And the Japanese are, you know, it's an island and they have lots of cool toys for mapping offshore. They had just done bathymetry mapping, high resolution bathymetry mapping of the toe.
Wow.
So the thing, you know, where the plate's going down, they had just mapped that area. And then they went back after the earthquake and mapped again and were able to difference those images because they had such high resolution. And that's how they knew that the toe, it moved like bloop, it moved like 50 to 75 meters during that earthquake, which is why they had that awful tsunami. Because that much movement is like that much water just went up and down. And now you have this devastating tsunami. But that's like the only place I know of that they have like before and after to be able to say that, because it's really hard to say much about like what's changed in unless you have really high resolution bathymetry. All right, we're good. My time here is done. I don't know who's up here, but hi, Jamie's coming. Okay, what's that? We're ready to take up the heat flow probe. Thank you all. All right. Back to biology. Thanks for listening, everybody. I'm not sure who's coming up here, but I think it's Jamie. And I think we're starting a biological transect. Cool.
No, I do not. Yes, I do. I do. Hello.
How you doing? Okay.
Yeah. Oh. So since we've been at the same spot for a while, we are gonna just move around a little bit for like the first 10 meters or so, looking around, and then we want to start a transect on our way. All right, good morning, everyone. I will be here just for a very short while before my colleague Eugenia takes over, but we are ready to start exploring. So they just took some push cores and heat flow measurements, and today our main goal is exploring this mound and looking for any sign of bubbles or seepage or high density communities. And this is an area that has never ever been visually explored, so it's very new and very exciting. And so right now we're sort of like in the bottom of a valley-like feature, and we are about to start climbing to the top of the feature where we did see some bubble streams mapped last night. So now our goal is to find them. And hopefully if we do find it, it'll be super exciting like yesterday, where we should see lots of chemosynthetic communities. But you never know. That's the beauty of science, is we never know till we're there.
Is this a transect or we just fly?
Not yet. We're gonna start transect soon. We're just looking around. excited? Yeah, super excited. Yes. Not yet. I'll let you know. We're just taking a little quick look around, but we're about to start one.
You can get like 1.5 high.
It's a brittle style. We might as well start trying to
What?
Yeah.
It's 0.2 speed, right?
Yes. Point. Yeah, so sure. 0.1 meters per second.
0.1?
0.1 and 1 to 2 meters above the sea floor. I think 1 should be fine.
1.2 meters.
1, sure. And then when we begin, we just flash the lights so we can break up. And then every 100 meters, every 300 meters, we'll flash the lights again just so we can break up the segment. You never know what to do. So, I mean, if we do see something exciting, we can stop and zoom in. It just means that when we zoom back out, we need to go back to like the exact same position. Yeah.
We didn't start yet, right?
No.
No.
Transect at the 1.2.1 speed. Need the camera, Zach?
Download.
Yeah, sometimes I realize the laser dots, like one of them disappears.
Because of the,
Angle.
it's the laser right under the cameras, right?
Yeah,
Because
the
now
two
it
green
needs
dots.
something.
Are we able to angle it so we can see both?
Yeah, when we lift
When we
the
lift it up.
camera.
Oh yeah, he's fixing it.
Where's the camera? On the left.
Did it.
Or did it?
One meter. Can start.
How we're starting the transect. 300 meters. So in Trinidad and Tobago, our deep ocean makes up about 70% of our marine territory. And we have actually visually seen less than 0.001%. Even though it's our largest habitable space, it's entirely unexplored. So we have chosen a variety of spots that have different depths and are spread across our marine territory to begin exploring. Again, this is hopefully the first expedition of many, because we know very little about what exists down there and what it looks like. We also need to be able to map our sea floor for when we do these dives, because we can't really go down too blindly. It also helps us pinpoint areas like where we see bubbles on the multi-beam mapping. It pinpoints that there may be methane seepage there and that high density communities may exist there. And so that's why we are focusing on these areas, because again, we can't protect what we don't know or understand. And globally, the deep ocean, because it makes up the majority of our ocean, plays a huge role for humanity in keeping everything on Earth alive. It absorbs a lot of carbon dioxide, it cycles nutrients, it's home to many of, all the fish that are important to us for fisheries and for food consumption. There are possible medical benefits. And intrinsically, it's a home to many animals. And so this is just the very beginning. And in Trinidad and Tobago, we're looking to help provide the information and knowledge needed so that those with the ability have the knowledge they need to make informed decisions when it comes to how we utilize our ocean. Otherwise, we're just making decisions blindly in the dark. And so while we'd like to see our whole
go ahead.
ocean floor,
Point.
You wait.
we have to begin somewhere.
They were running at point 2.
And we have 1 point here, but I'm thinking maybe 0.2 would.
This is point 2.
Yeah. So we see that there are bubbles. Should be within each there, but this is what we've never seen before.
Mm-hmm.
Okay. Seems a little faster.
It's point 2. This is
Well, I think we'll leave it. I don't know. So we're going at a speed of 0.1 meters per second because that was instructed on our plan. But it feels very slow. Like maybe it should be 0.2.
This is point 1?
Yes.
Yeah.
No.
Can we go to 0.2? Yeah, it's a bit slow. Now it is. Yeah, this is point 2. I'm gonna make a note on our map plan
This one.
that okay, well that was a fun 15 minutes. I'm gonna hand over to my colleague Eugenia. Alright, thank you everyone.
Thank you, Jamie, for your narration. Hi, everyone. My name is Eugenia Thomas, and I will be narrating for the next hour. I am a marine biologist specializing in deep sea biology and also a MPhil candidate at UE studying deep sea woodfalls and deep sea debris of the Caribbean. And as Jamie has mentioned, we have just started our 300-meter transect. So this transect is of a valley-like feature, and this dive will focus on locating the bubble streams on which we had mapped yesterday. And so we're just making our way.
100 meters.
And so we are just heading along the ridge-like feature in an eastward direction. We have just passed our 100-meter transect line, and we're trying to locate the bubble streams that we have seen in our mapping earlier. And so right now we're seeing sedimented seafloor, but yesterday we saw some methane seeps. And for those who have just joined on, methane seeps are one of the ecosystems within the deep sea environment. So what happens is that organisms or organic matter over time will decompose within the sediment. and eventually that decompositions develop methane. And over time, the methane in the sediment will become heated and pressure will build up. And that pressure causes the release of methane through cracks or fixtures within on the sedimented seafloor. And with that, you have a lot of bubbles coming up, what terms as bubble streams. And so that's what we're looking for today. We also saw a lot of carbonate rocks as well, which is another result of methane seeps or methane within the sediments. The lasers you're seeing on your screen are at average 10 centimeters, and this is what we use to give a measurement of species or ecosystem.
Hmm? I don't understand.
I haven't seen much comments rolling in. Like it was like in this room for like a while now.
Mm-hmm.
You can't try to do
Normally you just see like
Scroll
comments. Yeah. I find strange. Like nobody says hi. I don't know.
It's working.
It's okay.
Tugging it around a little bit now. Tugging it around a little bit.
Yeah, big.
So far we've seen a lot of sargassum on the seafloor. And as you know, in the Caribbean region, we have high amount of sargassum on our shores and on the surface waters as well. Oh, what is this? It's a sea cucumber, holothurian. And sometimes when you see these organisms on the seafloor, they leave tracks. And so that helps us determine, if there's any animal that would have passed by earlier. So even though sometimes you don't see a physical animal present, sometimes observing these tracks can help you. It gives evidence of such. And likewise, even though you don't see an animal on the surface of the seafloor, there are so many small organisms that are in the sediment as well. So sometimes polychaetes or worms or other crustaceans will bury themselves in the sediments. Absorbing organic matter or feeding on any small microscopic organisms that are there. There's a small little jellyfish just past us. So I've shown you this many of times, but just to reiterate, this is the first locally led deep sea expedition in Trinidad and Tobago. We had a exploration a couple years back in 2014, where a few scientists were able to discover and explore methane seeps. And so this expedition is really, really important because the samples, majority of the samples that we collect here are going to be stored locally at the Zoology Museum at the University of the West Indies St. Augustine campus. And so ultimately this expedition was created by us for us. All the sites that we have chosen were designed to help guide policymakers and really just to show the public, especially, what exists on our seafloor, what species exist, the behavior, characteristic traits of animals. This is like providing a baseline or species database to show that there is life within the deep sea, especially within Trinidad and Tobago. And so this really set the stage of better protection and conservation of our ocean space of the deep sea, because the deep sea environment is our largest ecosystem in the country, but it's the least unknown. It makes up 70%. And so this is really to start management work. So we just passed our 200 meters in transect. In total, we are doing 300. And see some corals here as well. Saw a lot of these sea urchin, starfish. A lot of sargassum is near the bottom of the coral as well. Could you zoom in on that line? Yeah. So I think that is a, looks like a fish line. It appears like a fish line. Yeah. And so we've seen a lot of trash on the seafloor. From garbage bag.
You're still moving, eh?
RV Bridge, go ahead.
Are you still moving?
Sorry, can you repeat that again?
Are you still moving?
No, is there another waypoint?
Oh, sorry.
Okay.
Okay,
Okay.
there you go.
I'll keep moving.
So we've seen a lot of trash on the seafloor, and this is just an indication of how our activities on land impact the deep sea environment. Whether it's from poor waste disposal or even abandoned lost or discarded fishing gear. You know, these items sinks all the way meters, thousands of meters down to the seafloor. And so what usually happens is you have a lot of negative interactions with debris. So sometimes fishing lines or plastic bags can entangle coral colonies or sponges, and these organisms, these habitats are prone for entanglement by debris because of their branch-like structure. So sometimes these fishing lines can snag on the branches, even cause tissue loss, or in some cases even death. Sometimes trash can provide a substrate for animals to grow on. So sessile organisms like sea anemones or corals can adhere themselves onto the surface of a debris. But what usually happens is a lot of negative interactions. So it's really, really important that we recycle, dispose of our waste, our items carefully on land and even out at sea. A lot of many corals. Here's another one of our favorite fish species that we have seen on this dive. So there's so many organisms here. We have two different types of corals. We have some brittle stars. You have some sponges, encrusting sponge. And so sometimes when you see these rocks on the seafloor, they don't just, exist. They provide a substrate for many organisms.
We have to finish it. So. There we go.
Shrimp is really wanting attention from us. So earlier on the dive today, we collected sediment cores. And these are used to basically, the experiments or projects can come out of this, such as looking at the macrofauna abundance or the meiofauna, which are smaller organisms.
Can you go parallel to the line because we're measuring distance?
Oh, parallel.
Over there. Seems a bit off in the transect, so you should parallel.
Can you go parallel to the line? So earlier on our dive today, we collected sediment cores. And we also did heat probes as well.
Difficult right now. Yeah, it's turning me.
Remember it was a minute ago it was bucking, or did it stop?
I felt like after pulling up the cable, it got worse.
The current strong.
Just turn it on, so we can turn it off.
Trying to figure out.
All right, you told him already it's past 300.
300 plus 300 meters.
Okay, fine. transectors.
Trailer jar, the updated waypoint.
Updated waypoint.
yesterday? Yesterday you been here? No? Yesterday was good. Apart from this, Mm. I never. I would like to.
We saw one shark.
Oh really?
Yeah.
This trip?
Yeah.
Really?
Yeah. I think it was 3 days ago.
Oh nice.
Yeah. So hopefully we see another one.
Cool.
We've seen a lot of holes in the sediment as well. And again, a lot of animals tend to burrow themselves in the sediment. On other dives, you also saw some sargassum, a heap of sargassum or sargassum bed. And you saw a lot of tiny organisms like amphipods that were in the sargassum, you know, just absorbing all the organic matter. And then other organisms will come feast on amphipods, and that creates a whole food chain basically. This is a very, very long animal track, likely made from a sea cucumber. You see that long vertical track that is from an animal. We also see in the fecal cast. Again, this is evidence of life. Yesterday we also deploy some colonization experiments. So we deploy both wood and bone. And so one of the goals for this expedition is looking and seeing what animals colonize wood and what animals colonize bones. And so we're deploying this near on active seeps and far away from active seeps, and just basically comparing species, what exists on wood and what exists on bone, and then we have common species, and looking and seeing if the species varied from on active seeps or far from active seeps. And so we have one more deployment to do, which hopefully will happen Friday or yeah. And so these are experiments, but they're in the deep sea. They are natural wood pieces or animal carcasses, and this is termed organic falls. And so it's what the name describes is organic material, so branches or tree logs from on land that makes its way into rivers and stream and into the oceans, where they could become saturated and sink to the seafloor. Or it could be animal carcass like whale bones or manta ray, which we saw in our previous dives. And so these type of organic falls create a habitat or a substrate for animals to feed on, and they usually start with organisms that are adapted to degrade the material. So for example, on wood, the xylophaga species are adapted to degrade the wood into wood chips and organic matter or carbon into the surrounding water column. And then on whale carcasses, you have an organism known as the ostodac worms that are also adapted to degrade the bone material. And so these are called specialist organisms. And when all this degradation happens, it can attract many other organisms, and these are known as opportunistic organisms. And over time, when you have a buildup of wood chips or bone material in the water column, sometimes they produce sulfide or sulfur in the environment, and that attracts many bacteria known as chemosynthetic autotrophs, bacteria or microbes. And sometimes these can last up to years, decades. And so they are both natural organic falls as well as experimented falls. within the deep sea. A beautiful crinoid. And so yeah, so organic falls basically shows you that even though it doesn't originate within the deep sea, it can come from surface and provide a home for organisms. And it really helps challenge the argument that the deep sea is food poor, which it isn't. It have so many life, so many organic materials that comes from land or surface of the waters that provides this food bonanza to the seafloor.
It is holding back.
Oh yeah, Jonathan.
Are we rich?
What's your current speed?
Current speed 0.4 over the ground.
Can we try a 0.6?
Yep, come on, 0.6.
Thank you.
Hmm? Wow, amazing. It's my first trip.
That was the
Road to it.
Yeah.
It was amazing.
I think it was a doldrum.
Yeah.
Yeah. So those images, it looked really nice.
And I never heard about hydrogen before. And then my first trip is like so
I mean, we see some pretty cool things and all that. As well. You see like the manta ray photos. So many of you have been asking what's it's like being on a deep sea expedition. First of all, it's amazing, hands down. I think every aspiring deep sea biologist would want to be on a deep sea expedition. You know. And as students or early career researchers, it's one thing to write papers and look at ROV videos, but when you're actually on the expedition, you're seeing things happen in real time. You're seeing the exploration happening. You're seeing the species that are being discovered. You're literally interacting with so many different scientists in the field. So it's really, really pretty cool being on an expedition, especially in Trinidad and Tobago. So this expedition is a month-long expedition, and we have so many objectives, from cryopreservation to organic fall experiments to mapping the seafloor, really providing that baseline data for our country. For those of you who are interested in a career in deep sea science, there are many different avenues you can take to become a deep sea scientist. And there are also many different fields that you can venture in. So if you want to focus on coral reefs, there's also scientists that focus on organic falls. There are ones that focus on the chemistry environment. There are ones that focus on energy, and this can also be in the form of like chemosynthetic habitats. You can also do mapping as well. You can focus on rocks, so like a geologist, a deep sea geologist. You can also do image annotation, which is really, really important. There's also science communication, translating scientific findings to the general public, whether it's in form of social media posts or ship-to-shore events, which is basically a virtual event where we live stream life on a ship, life on an expedition.
Okay, waypoint.
And so actually prior to this expedition, we would have done a lot of school visits, just setting that stage for students and for the public to be aware of, what is happening, what does it mean for our country, our economy, our environment. So again, for those who are now joining us, we are coming live from Trinidad and Tobago, and we are locating bubble streams that we would have mapped earlier. So this is our third site, and we're seeing a huge trash bag. Yeah. And if you look around the trash bag, oh, there's actually a fish in the trash bag as well.
Inside, man.
Yeah. Fish. And so he or she is trying to feed off of any small organisms that are on the trash bag. And so I could imagine what other organisms are inside there as well. And you can see the inside trash bag is providing a shelter for organisms. So what I would have mentioned earlier is that sometimes trash can be positive in the deep sea, but it's mostly negative. So sometimes trash bag can provide a substrate for organisms, sessile organisms, or it could provide a shelter. And then we have a sea cucumber. Looks quite different from the other sea cucumber that we see. And if you look closely, you see a sort of circular pattern around the trash bag. And so you can actually see where the sea cucumber would have moved. So it moved in a circular pattern, so that is the tracks of the animal. So this guy moved around a lot. Probably was looking for, what organism he could feed on from the trash bag. This sea cucumber is very, very much, it looks very different from the organisms we've seen. And yeah, and then the bags. So the circular pattern is actually from the bag. So there are many currents within the deep sea. There are downwelling currents, upwelling currents. And so the currents now, this current, say, will have caused the bag to move in a circular pattern. And so this pattern can also show you the direction of the currents, basically. And so this would have been moving clockwise, or even anti-clockwise. But it's been moving in a circular direction. So yeah, thanks. And so this is the many ways on which currents can help drive or determine, the behavior or any other characteristics, traits in the deep sea. So for example, near seamounts, and seamounts are underwater mountains, there are a lot of downwelling currents, so currents that would have moved in a downward projection. And this can cause a lot of species near or at the bottom of the seamounts. And seamounts are actually said to be a very diverse ecosystem because of that downwelling current you have. So a lot of organic matter or particles or microscopic organisms would have been directed or funneled in a downward projection near seamounts. And so likewise, how the currents would have moved the trash bag in a circular direction. Sometimes what you also see, and I've also seen this myself, is that currents can move trash bag from one location to another, not just in one spot in a circular direction, but it basically moves along the seafloor. Little small coral near to a sargassum. So yeah, I could just imagine what other organisms were using the bag as a shelter. I suspect that they are more than just a fish. And what can also happen too is that you can also see eventually over time you might see a sea anemone on the trash bag. So again, we really need to be very conscious on how we dispose of our garbage on land or even out at sea. It reaches the seafloor. And trash like plastic can't exist. It takes long to degrade in the deep sea environment because of the cold temperature. So, yeah, we zoom in on those two things. It looks like, so this looks like a mussel that has been opened. So this can also be an indication of, and there's also more up ahead. And these type of shells are very much associated with methane seeps. So sometimes looking at different organisms on the seafloor can give you indication on what's ahead. It's a nice sea cucumber. See, currents are really, really strong today, but we are powering through thanks to our amazing ROV pilots. And when we sit in the mission control room, you know, you are hearing us chatting, explaining about the deep sea, but it's really the ROV pilots that are really doing all the hard work. Like they're literally driving a minivan in the deep sea. So they are really, really talented guys, and we are really appreciative of all of them. And so if you're ever interested in a career as an IOV pilot, you know, we have some amazing ones on the Schmidt Ocean Institutes. So feel free to chat with them. And so they are driving the ROV Sebastian. And for those of you who don't know how ROV looks like, you can think of it like a minivan that is being driven from aboard the ship. And a AUV, a HOV is different. This is where a human will occupy the vehicle, whereas a ROV is being remotely controlled. And here's another one of the sea cucumber that we seen earlier. Very much look different from the ones we seen earlier, well, from our previous dives. Again, these lasers are on average 10 centimeters apart, and this can be used to help measure organisms on the seafloor. Oh, that's a huge starfish above us, right ahead of us. I think I also see a sea anemone. Wow. And the color too is also amazing. And there's a very small shrimp swimming along, trying to find any small organic, small microscopic organisms. There's a big one and a little one. And this starfish is really, really amazing. And you could see the little spikes coming out from under the starfish. So that's called tube feet. And they use this to move along the seafloor or on structures like corals here, as you can see. So this is an octocoral. This little shrimp is just swimming along. And shrimp looks, what is it doing? Is that, yeah, I think so. So my colleague Kyle just points out that it could be eggs. It looks like eggs on the bottom. Can you zoom in on it? Yeah.
Yep.
Yeah, definitely eggs. And the color of the shrimp though, it looks purple. And then there's this small little one that's constantly swimming around. But yeah, that's definitely eggs. Don't know if it's ready to lay the eggs, but it keeps doing a sort of upward movement. Actually never seen one with eggs. From all the RV footage that I've saw, never seen one with eggs. So this is really pretty cool. And there are many, many eggs. So these organisms can store many eggs as possible. This little guy is, oh my god, he's the cutest. What is he doing? No, really, what is he doing? So it's like it could be like he's just like waiting around, hoping that, you know, he find any small little organic particles that he could feed on. This is pretty amazing. I've never seen this. Again, you know, it's not just looking and seeing what species are there, but looking at the behavior. And this is a prime example of, you know, the purpose of our expedition. Thanks, guys. Yeah. That was amazing. I don't think.
No, we're just talking
Oh,
to them, sorry.
Good, because yeah, yeah, because the next one will be the third one that we see.
Yeah.
Yeah. So hopefully we see our other secret people like that.
The purple one?
Yeah, because that would be our third one.
Okay.
Yeah. Yeah.
We can collect it.
Yeah.
Yes.
I'm still in awe of that shrimp we see with the eggs. And everyone in mission control room were just blown away by this. And so we're still exploring. And here's another soft coral. Hopefully we could see another one of the sea cucumber, because that looked really, really amazing. Definitely different from the others that we have seen. And I wonder how it looks moving through the water column. Here's another starfish. Similar species to what we seen earlier. Ooh. Oh, it's a huge sea anemone. One really, really nice. Could zoom in on it a little bit. Could you take off the lasers? Oh, that pattern is lovely. So it's a little over 10 centimeters from the ROV lasers that were on. And this pattern is so beautiful. Oh my gosh. And the colors as well. Yeah. I mean, it's almost like if it's a ring pattern, but then there are other small patterns within the ring patterns. This is really, really amazing. Not sure if I've seen one like this. Could be, but you know. Yeah, we haven't seen one like this. This is really, really gorgeous. And pretty big. So guys, it was really a pleasure chatting with you guys. It has been an amazing hour, but I will now pass you over to an amazing colleague of mine, Charlotte Tom, and she will take over the next hour. And I will see you guys soon. Thank you.
Good, thanks. Thanks, Eugenia. Hi, everyone. This is Charlotte from Trinidad and Tobago. I'm a member of Species, and I'll be taking you through the next hour of this dive. So, right off the bat, we're going to take a closer look at this sea cucumber. This is the same one we've seen a few of so far. Could we put the lasers on? We're gonna go ahead and put the lasers on as well so we can get a good indication as to the size of the sea cucumber. He looks like a big boy. So can we just confirm we are gonna take this guy as a sample. So might be a bit of a challenge. So
Probably try and gently grab him, but he might be too big for the jaws.
so right now our ROV pilots and scientists are just working out a method as to the best way to collect this.
You're gonna have to go in the bio box,
He's
right?
very fragile, very soft.
So video with lasers on, video with lasers off. So it's all in there for 3 seconds, and then pictures, and then pictures. Ready?
Wow, look at that texture. So we're gonna take some videos of this guy with the lasers on and some with it off, and then we'll get a couple close-ups as well. Wow, look at him move through the sand.
You need the laser on?
Is it here? Did he get some screen grabs of this one? Cool.
Oh, it's a
Oh,
swimming.
wow. He's coming closer.
Open the box.
Yeah. Maybe he'll open the box and jump in.
You can see that well.
He's moving very graciously away. How are you guys gonna collect him?
I think we're gonna grab it. We're gonna grab it.
Might have to
Yeah,
back up a
with
little.
what? The arms?
Mm-hmm. You need to jump back, right?
Yeah, jump back over.
Wow. Let's see if we could very carefully pick this guy up.
Is the speed all right, too?
Right, moving.
So jello.
Wait. Oh, well, no.
Jello.
Well, as you could tell from the lasers, which are a good size and indication, 10 centimeters apart, this sea cucumber is huge. Also, the first time I'm seeing one like this. So it's very exciting.
Even just touching this guy with these rubber tips is kind of tearing him up a little. Can I poke him a little?
A little. Let's see. I don't think it's that soft.
Very gently, yeah. Oh.
He's just falling apart when I try and do anything.
Could the claw one be bigger or not?
The tip is more flexible from the
He just falls apart. It's like gum.
Yeah. Is it possible to open the claws even bigger? Yeah,
the
that's a
one?
grab.
No.
No?
That's the biggest they go.
That's the widest they could open the claw.
Scoop.
Is it too big for the net?
He can be too big for the scoop too, but we can try. Go ahead and porch out whenever you got a good view of him far away.
So we have a number of different ways and methods that we collect these samples with this ROV, and different storage boxes that you can see. So right now we're trying to figure out the best way to pick this guy up, based on his size as well as being a soft-bodied animal. Yeah, we're gonna try see if we can try the net with a scoop.
You want to try the net?
Yes, please. And net or scoop, which you think is better?
I think net would be better. Probably.
Yeah.
if you need.
So right now we're gonna try to use the net instead. Our very skilled ROV pilots maneuvering to see if we can collect it. So sea cucumbers are often called the vacuum cleaners of the floor because they feed on sediment and digest the organic matter, often leaving behind cleaner sediments. So they also play a really important role in recycling nutrients and ensuring that the deep sea ecosystem remains healthy. So as I mentioned, this is, at least for me, the first time I'm seeing a sea cucumber like this. And he is massive. Oh. Very skillfully dodging.
Too big for the net. Feels like a turkey.
We can scoop with the firebox.
Maybe. I mean, I can try and hold him up and then drop him in there, maybe.
Yeah.
Just,
Or
yeah.
maybe just like hold them
You're
steady.
just gonna have to go straight up, real gentle once I get him. You know?
Right.
Nope, not yet. Just get
Not
down
yet?
there.
Right, no. Sorry, they
I'm
can
gonna have to get him above your porch before you porch out. You know.
Right.
Mm.
Straight up. Nice and easy. Straight up.
So close.
Going up right.
Ooh. Look at green.
Straight up. You can porch out if you want. Go right. Right. Right up.
We're trusted up here.
There it is. It's really playing ping pong with this poor fellow.
Yeah.
Get back up if he's too close though, okay? Can't come in close. Oh, oh, oh.
I think it will fit in the mailbox.
You got much else to show?
As you can see, it could be quite challenging collecting these samples, to say the least. Where did they go?
Is that mailbox one?
3.
We're hoping for. Alright, so if you go forward and I open a little,
Yeah.
we're going there. Ready?
Going forward.
So there's a lot of teamwork and coordination happening right now to make this happen.
I just don't know if he's gonna fit, yo. One more left.
Going left.
That never is going to open again. That's it.
Did you get it?
It is there.
Is it? And that's BioBox
3.
3.
3.
Okay, great. Big up our ROV pilots once again.
But we can't reopen that now. Ever.
Seep open. On the left, one seep on the right, or something.
I don't think we should open that again. Oh, yeah, we know we're not gonna do it everywhere.
If we open it again. Yeah.
Okay, do this. That was wrong. Camera.
Thank you. Three. 3B. So that was actually our first sample of the day. Yesterday we've had lots of samples. And collecting these samples is really just one piece of the puzzle once Sebastian comes back up. Once Sebastian comes back up, then the processing really begins. And we have to ensure that we take really good care of these samples, take good photographs, and then they're off to preservation. So I'm now being joined by a new shift change, a new set of ROV pilots here. There's also a couple other scientists in the room, as well as Zahir, who I'm sure you all remember, who's helping us with the logging today. So let's continue to explore and see what else we can find. I think coming up here we have a sea pen. Oh, it looks like it has a friend wrapped around it. Looks like a brittle star. So these brittle stars you see wrapped around the sea pen will oftentimes climb up, and as they're suspension feeders, they'll use their really long arms to reach out and see what they could feed on from the sea snow. Also in the room with me, there's our artist at sea, Marco.
Hi, everyone.
Who's been gathering a lot of inspiration for his projects. That's a really important part too about that SOI does and about this expedition. It's of course about the science, but also about communicating that science to a wider audience and using these live streams as well, for example, so that everyone or a large portion of society has access to be able to explore along with us and also learn about our deep sea. So it's been really great to see such a wide variety of nationalities represented in the chats. Also a lot of youth, a lot of students and schools tuning in. It's nice to see that it has a wide reach. We also saw a couple mussels earlier on while Eugenia was narrating, which is a good sign, a good indication of coal seeps, which we were lucky to see yesterday and previously. So hoping we see more of that as well.
updated waypoint bridge.
You waypoint.
Some more coral here, it looks like. Can we take a... Can we zoom in a bit, please? What kind of coral is that?
Take these. Zoom in.
We get it really close. So we're trying to get a closer look at this coral right now. I see a hermit crab in the back there. There's also, it looks like a squat lobster wrapped up in the coral. So it looks like a octocoral. We're gonna zoom in here and take a couple frame grabs and get a closer look.
Take it off.
Oh, there we go. We can get a real close look at all the detail of this octocoral. Okay, we got some nice frame grabs. So actually gonna take another closer look at this octocoral and see if we can get even closer to look at some of the details.
seepage, you could come to us.
So we are going to go ahead and take a small sample from this octocoral, take a piece of it. So as I mentioned, we're gonna take a sample, just a piece of this coral. And the good thing is corals are regenerative, so they do grow back. Similarly to the sponges that we've taken pieces of as well, they will also grow.
So you want to have that coat?
So these are our coral quivers here. We're gonna go ahead and take a sample and hold it in one of these. So we have our logger here as well, meticulously keeping track of all the samples and where they are, as well as all of the species that we're taking. Nice. Great job again by our ROV pilots, and another successful sample. That makes 2 samples for the day for today's dive. So for a few people who are just joining and wondering in the chat, the team of scientists on board, it's 25 of us, and 16 are Trinbagonian, and we also have a Bayesian amongst us as well. So I see some questions wondering if it's Caribbean scientists on board, and this is actually Trinidad and Tobago's first Trini-led deep sea expedition. Thank you. For those just tuning in as well, this is currently at our 3rd site. We are currently at our 3rd site of this Deep Wonders expedition, and I believe our 8th dive. We've collected lots of samples so far, and for this dive, secured 2 samples. We have a few more hours, so let's see if we can find some more. Closer look at that. Some more coral here. I'm gonna take a closer look. Looks like it has a fan of or a crown of bristle stars around this coral. Wow. It's really crazy how they just tangle up like this. So, as I mentioned, brittle stars are suspension feeders, so they will use these sea pens or corals and wrap themselves around it and climb to the top so they can get a better chance at extending their arms and trying to get as much food as possible. Wow, that's incredible. How many do you think are there? And what is this? It seems there's another friend there. It looks like they may have captured something. Yeah, so we're gonna zoom a bit more. It looks like they may have captured something and reading it in. Oh wow. Yeah, there's about it. What? It's like some sort of organism they've captured that's kind of curled up in there. This really beautiful coral here. Lots of stars on it. You can see some sargassum at the bottom as well. Oh, it's also really, really tall. So it looks like what we were seeing before was a bit deceiving. It's actually just the center of the brittle star. Ah, this is a gorgeous shot. take a closer look at this. It looks like it might be trash.
Just zoom in on the camera.
Camera, drag out. Camera to the drag out. Watch it then.
Okay, guys, thanks for joining us for the last hour. I'm gonna leave you in very good hands, handing over to Shirley, who's our sponge specialist. All right, thanks, everyone.
Okay, so we're heading between waypoints 2 and 3, is that right? So basically, we have a long way to go on the dive, so unless we see something really interesting, we're just going to keep on moving towards waypoint 3, okay? Okay. Hi, everyone. Back again. I'm Shirley Pomponi. A sponge biologist, biotechnologist from Harbor Branch Oceanographic Institute, Florida Atlantic University in Fort Pierce, Florida. And we are proceeding, look at my notes here, along a path that was planned out last night, early this morning, based on some mapping data. We are trying to get to an area where we detected some bubble streams during mapping, but these bubble streams have not been visually confirmed. So maybe that there are unexplored seep areas. Yesterday afternoon when I was on, I had my watch ended right as we got to the really cool part. Not that the rest of this isn't cool, but right when we got to the part where we saw lots of seep organisms. You stayed on to the very end. You saw methane ice. It was really, I mean, we all got to see it and enjoy the video footage and enjoy the dive. So it was very, very exciting. So we're hoping to get to a similar site maybe sometime during the next few hours of this dive. Right now we're on soft sediment, and we've seen the animals that we've come to know over the last week or so of our dives on soft sediment. Beginning of the dive, we did some push cores. We have scientists on board and back on shore who are interested in analyzing those for heavy metals, pollutants, and so on. At the same time, actually not the same time the ROV dive is going on, but right after that, and also right before that, we have a number of other activities that are going on. We do plankton tows almost every day, sometimes twice a day, for both phytoplankton and ichthyoplankton, so fish larvae. We'll be deploying a benthic lander called Doris later tonight, and trying to think what else we're doing today. I think that might be it. It's a lot. Our days are packed. We have very long days. Things are going on, happening 24 hours a day. So we have the lasers on. That gives us an idea of the scale of things that we're looking at. Those lasers are 10 centimeters apart. They're useful not only for us now as we're exploring, but also as others will be using these video tapes in the future to identify, quantify what they're seeing on the bottom. We've seen quite a few of these sea urchins, collected a couple, I believe. I know we collected at least one. If you were on earlier, you heard Dr. Rachel Lauer explaining some of the work she's doing and the experiments she's conducting while she's out here. At the beginning of the dive, she collected some heat flow measurements. She can only put that probe in if the sediment is fairly thick, so she had that opportunity at the beginning of the dive. Still seeing some sargassum that has settled down from the surface of the ocean. It's getting a little rough out there. Seas, I think, are expected to build even over the next day, but we're on a big ship, so we don't feel it too much. Like another octocoral coming up. Just keep on going.
Rich, you could continue with that.
actually moving towards an area that looks like it's a kind of a, I don't want to say, like a depression, a basin in the bottom. That'll be where, I think that's where our next waypoint is. So we're seeing a lot of tracks on the sediment. We're seeing some small depressions and larger mounds, and those are all created by animals that are living in or on the sediment. Like another sea cucumber, similar to the one that was collected earlier.
that, Julie? Is it a dead mussel? Someone
We're
said that?
seeing,
Yes.
we're seeing some shells. That's
Good
a good
sign.
sign, right?
Yep.
Good sign.
Yeah, go.
I'm gonna get some photos on this on my watch. So we're headed towards waypoint 2, and it's an area where the multi-beam mapping showed some evidence of bubbles. So we're hoping to see methane seep communities in the not too distant future, moving along towards waypoint 2 right now. You can see those bubbles on the multi-beam. Juliana, can they see the map on the live stream? Almost there. Seeing more shells now. Anna. You wanna just head straight towards waypoint 2, or you
Yeah.
wanna kind of move a little bit
Well,
over?
it's hard to tell because of the noise. So what you're saying is.
Yeah. All right, so we should go right through it. Right, we should go right through it. Okay. All right.
When we get the 2%,
Yeah. Yep. Sounds good.
That's all there is.
Yeah.
Depth is 1,391 meters. Our dives are usually about 12 hours long, so you take into account, the time it takes to get down and to come back up. We usually have about 8 or 9 hours of time on the bottom, which is pretty amazing. So we try and cover as much ground as possible. Again, this is an area that hasn't been explored before, so any data we're getting are new data.
What's that? Yeah, something, yeah.
What's that?
Yep,
Oh, yeah.
yep. Yep. What is that? I don't know.
50 meters.
Yeah.
I will intercept it.
desert, so.
Definitely seeing more shells.
There's all the stuff. Sponges. Yeah, look to the right.
Okay, so
The gravel mat on the sediment, and I think that might be. Hold on. So see, there's a lot more over here on our right. What's wrong? White mat, the white, so close. Yep.
Yeah, we're getting there.
Yep.
It's interesting, the sonar isn't really showing
It's not sure.
much.
all right, so we're starting to see the bacterial mats. Very much indicative of methane, seep habitats, more shells of dead mussels.
Yeah, I know. Yeah.
harder return.
Oh, look at that. Losing it a bit here, eh?
There's another return in the sonar.
Yeah, but it's to our
It's kind of along
It's to
on
our
the edge of
right.
that, yeah, that basin.
But again, like we were right under this before, I really wouldn't emphasize
No, yes,
this.
then yes. No, more looking at this
Yeah,
side.
the big clear, right?
Seems to be more.
They want to look at this, yeah.
Yeah.
The sun is shooting.
Yeah, okay. Just want to explore around this area.
to waypoint 2?
Yeah.
You want to get
Yeah.
to this on the sonar?
Yeah, we want to try and stay and, explore around this site. So, we can head a little bit further towards waypoint 2, but we want to circle around and look in that hole and see those. See what this.
So if you want to explore in here,
Yeah,
as part of
yes.
the tour, then I need the vessel to come.
Yeah, okay, that's fine.
You want to come here?
Yeah,
The vessel
yeah,
needs to,
yeah, that's good.
I need to look where the vessel is sat right now.
So we're right along the edge of an area where with the multi-beam mapping we saw evidence of suggesting bubbles. So we're going to explore around this area for a little bit. hexactinellid sponges. Stalked ones that are similar to ones we've seen before. Hyalonema is their scientific name. It's big.
That's huge.
It is huge.
Yeah. Oh, well, that's just the top there.
They want to look into the bubbles in the.
Okay.
So that's where the ship is sat right now, right?
Not necessarily. This is just, I almost want to
Yeah, we're just going around it. It's not. It's just a target.
Here, I'm gonna remove it because
Yeah, just remove it.
it doesn't necessarily mean the bubbles are there. It's
No.
just helping us pinpoint.
That's what
So
the
we check off
waypoint does.
sonar targets and waypoints, yeah. Because last time we went right under it. So could be like an offset.
Headed back to 2?
Yeah.
Headed back to 2? Good, you could come to us.
Oh, two
It
bugs.
looks like we're coming up
Is that two bugs?
towards that
Oh, looks like it.
edge into that basin.
Yep.
Yeah. Looks like some dead worm tubes. So we've collected probably about 10 different species of sponges during this trip so far. I think I have enough now that I'm going to do some spicule preps tomorrow. The way, one of the ways we can identify sponges using morphological characteristics is by looking at their microscopic skeletal element, those things called spicules. And we collected 2 different species yesterday at the seep, and I'm really curious. They're very different. I'm very curious to see what they are, so hope to report back to you. If it's not too rough tomorrow, I'll do some spicule preps. I'm not guaranteeing I'll be able to look through a microscope, and I'm going to try because I'm really curious. And then a few days ago, we collected what might be a carnivorous sponge, not the big mat that looked kind of like spaghetti, but there was a like really tiny. I don't even think they were a centimeter tall. Tiny sponges that kind of looked like, I don't know, like Q-tips, cotton swabs. And I want to be super careful with those because if they are a new species, we only have a few individuals of those. Make sure that we have enough intact material to try and identify that, whether it's a new species or not. Like around this,
No, just a minute.
just like around here, just zoom in on that. Okay, sure. Yep, okay. So we want to zoom in on this rock that we're seeing here. Yeah, see what's on there. It's carbonate rock, probably formed from the tubes and of these tube worms, and over many years consolidated by calcium carbonate, forming this substrate. Habitat for crabs and shrimp. So we're going to spend a few minutes taking a look at what might be living in or on these carbonate rocks. Definitely sponges, encrusting sponges, which would be virtually impossible to collect. Unless you picked up the whole rock, which is not be able to do that. Benthic ctenophore. Is there anything there that we want? The pink shrimp, yeah. We saw a few of those.
Yeah, so next one.
Okay, like that. Wait, not that one.
That,
No.
that one. That's
That
the
one?
right?
Is that the one? Are you stay? Don't move yet. Get some. May want that one. You can see little patches of encrusting sponge. Again, really difficult to collect. Is that the one? Is that the one? Okay. We may want to collect that pink shrimp. Yes? Okay, so, all right, guys, we want to try to collect that pink shrimp. Before we do that, we've already gotten some video, maybe get some more video of it before we go in for collecting it. The shrimp. Beautiful anemone, though. Okay. Could you just put the lasers on for just a few seconds, please, just so we get some sizing on that. Two. Oh, bottle two, okay. Okay, so has that already been cleaned out? One?
We haven't used
Oh, we don't need to? Okay, all right, so we wanna, in two.
Almost the flush.
Yeah.
So we'll flush.
You're gonna flush that and then put in two, okay, that sounds good. What we're gonna try and do now is suction this up. Before we do this, we wanna, clean out the suction tube. We do that first and then we'll suction up, try to suction this up.
So close up. With and without lasers?
Yeah, I did that.
You did?
Yeah, did that.
It's definitely easier to fly to. Okay, if you see my flush.
It's on.
Can you reach it from everything? We want to get a bit closer.
Say that again.
Sure.
Nah, you want it to go in a bit closer? You're good.
I'm good. Flush it first.
Mhm. Yep, good, okay, just flushed out the hose. Now we're gonna try to capture this really beautiful shrimp.
Yeah. Let's come down to the
That anemone is on the back of a crab. Okay, notice I said try. He had a different idea. All right, try again. Just keep a search image out for that.
Do you want to keep hold of it while we just have a look around?
If we see any of those, the team force, those red and team forces are attached, like red gel things, do one of those also.
Should we continue
I'll
to-
show you, I'll point it out too if I see it.
Okay.
All right. And you want to take a look more at those rocks, Hannah? You want to take a look more at the rocks, or start heading towards?
Well, there's the one up there, we can look at that.
Yeah, it's on the way.
Yeah.
We'll stop.
Okay. Yep. turtles, some crabs, sponges for sure. Keep going. Crabs really like this substrate. Waypoint 3?
2.
We're at 2. Well, you want to go the way up? Do you want to go all the way up to 2 and then?
Yeah.
Okay, all right. All right, we're going to keep heading towards waypoint 2 and look around. Okay.
We've got the vessel to come down this one, please. As you're moving, we're just going to have to walk up to
Virginia Waypoint.
Okay, it's my shift is over, and I'm going to be just turning over to Charisse now. I wanted to spend a minute or so just telling her what the plans are for the next hour. Okay, thanks, guys. We didn't collect them. I did see that. Oh, and then also, Dee would like the 13 for, I see that, and then we see the shrimp, yeah. Okay. Got it. Thank you.
Good evening, everyone. So I'm Charisse. I'll be taking over for the next hour. And as we explore, we're looking for seeps. But if we see any cool rock structures like we just saw earlier, we will stop and take a look, see if we find that shrimp again that has eluded us. And also if we find any of those benthic ctenophores, which were those red ctenophores that are on the bottom. And they typically sit at the bottom and they have these tentacles that are sticky on the ends, and that's how they collect zooplankton and feed on them.
So if we
Thank you. Thank you. Okay, so what they're going to do is because right now it's a bit to the left of us. Mm. Oh, can we zoom in on the skate actually? Oh, it's swimming away. Well, we're gonna reverse a little bit so we can actually get some close-up imagery of the skate. We've seen a few around the seep sites, but we haven't gotten much close-up imagery. So what we're gonna do is just zoom in a bit, take in some features to get some images for logging. Nice. It's so cute. If we could zoom a bit. Yeah. If we could get some zooms, I know it's trying to get. Look at it. You just keep frame grabbing. Thank you, Megan. Look at all those spines.
Take off this negative thrust there. This negative thrust is what's pushing this.
Uh-oh. Alright, it's on. Did we get some? Okay, no problem. Yeah. We got the images. Can we zoom in on that smaller one? Where's the pointer when we need it? Yeah. It's just resting on the bottom. Where'd you go? There you are.
Zoom in on that fish, please. That one. Whatever that is. It's trash.
It's on muscle. And fish. Okay. Got it. Perfect. Continue along to the rim. Do you want to put down the other waypoint? Looks like it. It's a good sign when you start
Look at that.
seeing old muscles.
The worms.
Two
Okay, here
rooms.
we go. This is good.
You can get some zoom on that.
Bridge, new waypoint.
Yeah, we're fine.
And a lot of bacterial mats as well. Yeah, zoom in on two muscles. That thing in the middle is
Muscles to your. You can't see them. Here.
Oh, yeah, I couldn't see that. Thank you. Yeah, we could get some zooms in the two rooms there, and then also the muscles. Whatever angle it is.
And bubbles. Bubbles.
Oh, we found bubbles! Yay! Of course it was on my shift. I'm so excited, actually. So this is what we've been trying to do for the last 3 days, guys. When they do the mapping at night, we see
That's a lot of bubbles.
these anomalies come up on the maps, and then we set up our waypoints so that we can look for them. And we have now successfully found bubbles. I'm excited to share that with you all. Oh, this is so cool.
I wonder if this is like a newer site. Since that's a lot of bubbles and not a lot of
I
animals.
know.
Where are the bubbles
Could be,
coming
and
from?
there's a lot of bacterial mat, which is
Yeah.
the first
Yeah.
stage, right?
Oh, okay.
So it could be.
Do you know where the bubbles are coming from? I have zero idea.
Oh, over there.
Yeah, let's take some video of
There's
those
actually
bubbles.
clamps on.
Here?
Yeah. But the current is going this way, so it's right there. See, it's running on here.
Yeah, so right now we're watching a bed of mussels. There's bacterial mats all around it, which are good signs of seepage. And we have a look at those
Yeah, let's
bubbles.
take some video of those bubbles if possible.
Come back out. Come back. Come back.
Yeah, it's emerging from right there. Stunning. Wow. Wow.
Come back some more.
Yeah, I might have to come back a bit. We're trying to come back.
Yeah.
Okay, no problem. Thank you. Yeah. Okay, cool. All right, so first we're having a position. We'll grab some videos here, and then we're going to do a temperature group.
our temperature.
So cool.
Look at that.
Look at snails.
And we're doing a video panel. I think they want to do so you press video panel.
Yeah, look,
Oh my God.
fantastic.
Look at that. And then you and then every one.
Elijah?
Wow.
Fungus creature. Bubbles.
We did it.
Amiable. Hey, love bubbles.
Just hold it in one area for a while so we can.
Yep.
I mean, you're fine wherever, but just let it be happy. Just let it,
Okay.
you know, that's just Monica will want to take up.
You can see it all there. Crabs are fuzzy.
Yeah.
Because they have all
The
these
back-
microbes in
yeah.
their mouth.
This is a lot of.
Look. We did kind of. Oh, look, look, oh.
Whoa.
There it goes. Oh. We did it, Rachel and Robert. All the mapping worked.
I've seen seeps where in every 5 minutes you get a single bubble.
I know. This is
This is
pretty
this is
accurate.
very impressive.
Yeah.
I keep getting spoiled on this trip. First time seeing a seep, and it's very active.
To the left.
Yeah, there's a good bit of bubbles happening.
How much further?
And there.
There.
Wow, look at this. Look at this.
Amazing.
Yeah, there.
Whoa. That's a lot.
That's two on a time. Yes.
The other,
So
the back
once you're
there.
happy with it, just hold it for like.
Yeah. This looks great.
Thank you.
No, after get videos with bubbles. So they want to do it, I think, right where those bubbles are coming, anything closer within the muscles. I'll confirm. But I think that's
Yeah,
all.
whatever you guys.
So we'd like to make some temperature measurements in the sediment, and above the sediment, and in it, and then by the muscles.
Can't believe I'm at the top.
There's another one. Oh,
It's huge.
bubbles everywhere. I'm surprised more people didn't run in here. I'm surprised more people didn't run in here. There's so many bubbles everywhere.
Everything's spot on right there. Wow. Waypoint.
There's so many bubbles. I didn't think it would be this many. In Valdo.
It's pretty nice.
Temperatures. Bubbles. All right, right. We're gonna do temperature measurements. We're gonna do one with by the bubbles themselves in that area, and then also within the muscles.
It's our temperature for water.
Ours.
Thank you guys.
Can I push him a little bit?
You just want to get it where the bubbles are coming up, and then also the next one in the muscles, where the better muscles are.
You want one right in front here? Let's point.
Do we have a pointer thing? Yeah.
Where these bubbles are coming up. If you can reach it.
Too much food.
So we're just gonna reposition the ROV so we can get closer to where the bubbles are so we can do the temperature measurements. So many bubbles. Now we know why they're not look at that. Makes sense.
Probably rotate. Come on the other side.
It's downhill. This is uphill.
Yeah, we're trying to figure out what
Other
angle to
side?
get.
Yeah, if I come the other side, then it's facing downwards.
So I think you would stay if it's that one. If you're gonna slide up. The reason why that side is because the arm is closer. If you spin on that side, the arm is closer to that side. This doesn't have much reach.
capture the original track. So,
Oh, there's the skate again. Are we heading away from it or to? Are you coming down with a compass?
just trying to find it then first.
Turn all this in. Skipped it. I didn't make it.
Yeah, you're right there. Can rotate. You're on it. Should we back in safe position?
No,
Stand
no.
back same position.
You want me to face downhill?
So that's why you left or what is that? But I mean it's up to you.
This is it here, right?
Yeah. Copy, copy of this. Huh?
One into the off.
Yeah, up ahead and turn fast.
This one?
You can turn it back one when you're off the road. Position. It's like the sun coming down from there. The edge of your porch right at the edge of the colony. Come on, porch.
I saw a singular bubble go up for a second. It's on the right side there.
Bubbles
You
right
see, yeah, bubbles. There you go. Is it right below us?
So you want to
Yeah.
add temperature, the clamps too?
So any muscles, the better muscles, and then also one where the bubbles are coming.
Okay, so let's do the muscles first.
Hold on.
1 second.
Read the message from Rachel.
For the tap measurement, be sure to keep the one out of the sediment so it doesn't heat up from friction. Okay, so for the temperature measurement, be sure to keep the one out of the sediment so it doesn't heat up from friction. Anna, can you explain that to me too?
Don't put it into the supplement, just above it.
Just above?
Just above the bubbles. Just above the muscles.
Just above the muscles. I guess
Confirming.
nothing had changed.
Where do you see the reader?
Oh, that's
Okay, Rachel's gonna come down and confirm with us. You got kicked out of the class now.
Rachel's coming.
Here you are.
Pick up the back. I mean, it's grade requirements.
I mean, you want this. I was giving you that. Also, there's.
Oh, excellent. 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 So, my father's obviously, but what do we have to have? It's the smallest.
You saw where the bubbles were, right? It's right on the what side, somewhere right there.
1 second, as I confirm how the measurement will be taken, because we're taking temperature measurements in the muscles and then also for the bubbles. So let me just confirm that. Coming down as well.
Yeah, we're gonna do it in here and then.
We're gonna do both the temperature probe measurement amongst the muscles and then where the bubbles are coming from, so 2 separate measurements. I just want to confirm how they want it put in.
Here.
Great. So Rachel is gonna come down and confirm with us how she wants us to take the measurement.
All right.
Hi, Rachel.
Hi!
Exciting bubble time. Can you come help guide on how you want that
Oh yeah,
measurement taken?
that looks good.
As in just like rest it there, but don't go into the center.
Yeah, because if you push it in, you're gonna frictionally heat it and make it seem like it's warmer than it is.
Okay.
And it might just be the same temperature as bottom water, I would expect, or colder.
Okay.
But I don't know.
So this is good. And how long do we leave the probe?
Just let it sit there for a little while and stabilize.
Okay. Alright, we just let it rest and stabilize for a bit. Thanks, guys. Why do I see the temperature probe? Oh, there. Got it. Thank you. Alright, so we just have our probe resting amongst the mussels. We're gonna leave that for a while to stabilize and then we'll take that reading. And we will do the same for the area where the bubbles are coming out from the sediment.
Dust. Oh yeah. That are really cool.
Oh yeah. You wanna just the focus or alter?
That is good. Oh! Nope.
They are.
They do look like they're pouting.
So amongst these mussels, you are able to see some squat lobsters, some snails, and then those kind of white-looking fish, Pachycara caribeum, which are like methane seep and hydrothermal vent dwellers. So when you see those, you know you found yourself a seep or a hydrothermal vent. Look at that guy. Adorable. It's been about 15 minutes.
So it's colder than Baltimore.
Can we make that image like this?
Yeah.
Yeah.
Up in.
Yeah, but
See, feeding their
that's-
lives.
okay. Can we write that down? No, come on. I don't know why.
Hey, why are you questioning my lawyer? Okay. We keep doing things if you want. But then after that, we're gonna consider bubble. Every bubble that, like, how do you get that? Yes, over the bubble. Okay.
Sorry? Oh, it's okay now, yeah. they need like 30 seconds for
Oh,
every
hold on.
answer.
Oh, I'm sure. Here's good.
Yeah, that's good.
No problem. Yeah, so basically what we're gonna do is put the probe over the bubble where it's gonna come from. We're not gonna put it in the sediment, just right above it. Is that for you? For you? Okay, thank you.
That's good. Thanks, Rafa.
I sip bubbles.
Right now. Time to go.
And we're just gonna put it right over where the bubble is coming out from, but not in the sediment. I think there's bubbles. Can you try and see? There were bubbles coming out of the sediment.
Out of the sediment.
Yeah.
Like in the.
On this right hand side of it earlier. If we could find the bubble.
Vamos olhar nessa parte branca assim.
I don't know if you see it anymore. Yeah, I see a bubble coming out. If we zoom a bit, maybe we can try and find that. Oh, this is done. Can someone also help me look for the bubbles?
Somewhere around here, the left-hand side of the crop, there should be bubbles. Right there? Down? Right. No. Right there.
You need to move more? Or it's okay there?
Or up and to the left side?
I think you can move it this left to this.
Just left to this color thing.
Around there?
Yeah, that's good. Perfect. Nailed it.
I'm gonna leave it for a while.
I'm gonna go there. It's too little to cause any heat.
That's not bad.
Is that one?
Mm-hmm.
Oh, yeah.
It is adorable.
Thanks, Brian.
Goodbye, yeah.
They're really big.
Oh, so do the chef. No, I was saying.
Is this what it's supposed to be? Look at these little sponges.
Yep.
Yeah. What time do you take? Which value do you take when it's not?
We're just looking at it. She wrote down that kind of an average.
Okay. Sounds good. Alright, so we want to just explore the area now.
Yes. Yeah, I think there's, I mean, even we just exploring this hole.
Yeah, that's it. So we got this point where we're gonna do bigger and bigger circles?
Yep.
Okay. Alright, so you can bring back in the probe. We got the measurement. What we're gonna do is, right now where we are, we're just gonna do circles, bigger and bigger circles around and just explore the area. See if we find more bubbles and seeps in life.
Cool.
Sounds good. Babe. All right, so we've taken our measurements and now we're just going to bring back in that probe and then do some circular patterns in this surrounding area and see if we find more areas of seepage and life.
We'll keep the probe with us. We're gonna measure more, probably.
Are we going to do more measurements? Should we keep it out or? No, we can put it back. Thank you. Are you so happy? I literally on the stream once. And we watched the journal.
Simples aqui, né?
I'm gonna have to do it Portuguese on this.
You should. I don't know how you did not run it yet.
How did I don't know. I don't know how I went my whole life without it.
Week here is what other language do you speak?
English, Polish, Spanish, no French. That's it. All I remember was nunca será.
So we're gonna go around.
Yeah, just keep doing exploratory circles, getting bigger and bigger. From where we are. Oh wait, can we double check if there's a waypoint at this exact location?
Yeah.
Yeah? Okay. Thank you. Just explored generally the area. Typically when you find seeps, you find live muscle beds like we just did, and bacterial mats as well, which is a glimmery, fluffy white thing. And those tubers, so that's very typical seep fauna that's happening. So as we look for more signs of life like that. And bubbles. Yes, right now we're in a big bowl.
It's the mark you wanted.
Mm-hmm.
This.
Is where we were just now, yeah.
Circle?
Yeah.
Portuguese.
Right. So right now we're in a bit of a bowl, and we're just gonna keep circling. That's where you see how they found. Oh my God! There's just so much bubbles in there. That's where the biggest stream was. So we're gonna search within and see if we find more things. It's going on the line. Was it inside just now?
Where we were.
So we keep. Oh yeah, you can see our sediment. So if we just keep circling bigger and bigger around it. Yeah. Turn right down there. Okay. Go into the bowl. Yes.
Yeah. Yeah.
Will he try to go around?
Yeah. So I'm just
She's
freaking out.
Where was the thing that we're over it right now? It's to the back of us? Just keep making a circle with this one. Someone is going to go with it, but we were already there. Just confirming. Still just do this.
Yep.
Yeah.
He was just very excited. We all are.
I saw it panicking. I was like,
You're good.
big shrimp?
Yeah.
Hey, buddy. He is pretty decent size, yeah. Traffic jam. Not answering that.
Circling around wherever you
Do we want to zoom in on the skate? Can we zoom in on the skate, please? The skate that we're in.
thing here.
Mm-hmm. That's a skate.
Skate?
Mm-hmm. Skate.
Skate?
So it's in the same family. Stingrays.
Oh no, you know.
I think it is throwing it back. Is that all the thrusting?
Yeah, because we got like the push cars,
Mm.
change a lot the weight, and then we get heavy.
Got it.
And then when you use the thrusters to go up,
Can we zoom in really quick?
you have a lot of force going down.
Ah.
That's why it's the-
Ooh. Stunning. Oh,
So we got like this balance check.
got it. If I have to retry and grab a shot. Look at that.
Ja, als controle kunnen we even voor.
The heart.
Dat wordt voor de rest.
Let me zoom out just slightly to get that. Oh, you said it. And then we'll put the lasers on after we get these close out. Wow. You're getting it?
No, I'm just sitting. Zoom in on its tail.
Zoom in on the tail there. Wow.
And they're alternating.
I just said it. Wow. Let me know when you got enough close-ups and then I'll put lasers on. Yes. So I will do one quick zoom up again and then zoom out, put lasers on. Sorry, I'm so close, I'm trying to do that. Wow. Can you zoom out now? Yeah. So we get the size reference. Ooh, don't swim away from me. Stay with me. Stay for the lasers. Okay, bye.
Whenever you're ready.
Okay, we begin our circle.
Only equal.
Isn't it?
Oh, she's there.
Oh, yeah, if we could take the measurement real quick.
That's good. It's like I'll put it in. Maybe.
All right, so I'm gonna hand you guys over to Shauna, who will be with you for the next hour as you search for more seeps within the area. It was lovely hosting with you guys. Glad we found what we did. We saw a lot of cool things within this hour, and I'm handing you over now.
Hi everyone, I'm Shauna. I am part of the research team on board.
Yes. Good to go?
Ski. You good to go? Yes. Yeah.
Just confirm within circles, right?
Yeah. Circles around the small seep. Yeah. Yeah. Circles around the small seep.
Yeah.
Down the slopes.
Exactly.
Yeah. So yeah.
So we don't want to get like far away,
Yeah, we want to stay kind of close by.
but explore a bit.
Don't go too far. So we're just continuing to explore around the small seep that we did find. See if we find any more bubbles. Yeah.
Yeah, we're looking for now.
Okay. Yep. Hmm.
I don't have much space inside there to do like bigger circles. See like that circle there?
Yeah.
Was around the this one like spanny for the top or just inside of it? No?
Sorry, say that again?
You're gonna check just in the valley or around
Yeah,
the road?
I think so. Kind of within the slope, within the bowl. Yeah, around the seep. So we're just exploring, seeing if we can spot any more bubbles, any more seeps, and then we may sample depending on what's available. So we're just checking. We're in kind of like a bowl, looking around, making circles around the seep, see what we can find. Another skate. I got the same one.
Oh.
Yeah. And C2. There's one. Two worms. Yeah. Yep. So we're just taking a closer look at some of these live tube worms. Ooh, those are long. Stay on them.
Yeah.
Let me know if you want more zoom. Yeah? Yeah, can we just zoom in a little bit more? No. You can see them retracting into their tubes. See some live mussels as well, maybe some sponge. Good to go?
Yeah.
Okay, you can continue on. Please. Yeah. And we will continue our search. Oh, another skate.
This is Juliana. Mr. Havens. There we go.
How many skates have we seen? How many skates have we seen? Okay. Yeah.
What?
Today's our last one.
Oh.
It's gonna be to the right.
Around. Okay, coming for around. So look at this slope. Yeah, another way. Can I take a yeah? Also look to that speed.
Also the fish. Yeah.
Yeah, it's large.
This is a muscle set.
Okay, can we keep an eye on
The fish?
Yeah. Yeah.
It went away.
Alright. It's okay, we don't have to chase them. We're looking skate. Sorry. Never looked doing boring stuff. Oh. And we think it's a male?
Yes, I made.
Yeah, it's a male skate. Yes. Down, yeah, thank you. Gorgeous. Can we get somebody lasers on?
It was on.
Yeah, they were on just now.
I just asked them to take it off for a second.
Oh,
Oh.
okay.
Yeah, sorry.
Can you read it like that, Rafa?
Gorgeous. Oh. Looks like he's gone under Sebastian, so we're just moving back. Ah, there he is. Oh no.
Hmm?
Can I get the lasers back on?
It's on right.
Oh, they're on. Okay, nice. Thank you.
The bingo.
Mm. Right. Can we get it zoomed in a little bit more, please? Oh, yep. I think that's good.
Since we are here, we can also zoom in on the crabs that have an enemy.
You've seen them?
Yeah, there's this
Around?
to the right side. Okay, just turn this next to the screen, okay?
You have more footage of the skate?
Sorry?
You have more footage?
Maybe like invisible.
Well.
I mean, it's kind of right.
Crabs.
Yeah, there are some.
So there's some crabs, I think a bit to the right, yeah, with some anemone, yeah? Yeah. Oh yeah. Can we zoom in on those crabs? Mm-hmm. No, no.
So bright, that one actually.
lasers?
Lasers on
Yeah.
this? I don't know if we're gonna get the laser there.
Zoom
We're
in.
probably gonna need to move this.
No,
Oh, yeah.
it's on there.
It was- Oh,
Yeah, it's probably, maybe you can zoom in without the lasers.
without the laser?
Yeah, it's fine.
Thank you. There's one under the
Is your donut crab as well?
top and then the
Good?
Yeah, thank you.
Anything else? Could continue in our circles. Yeah, I think we can continue looking in our circles. Please and thanks. So for everyone who's just joining, we're just doing some searching around, looking for some bubbles, some cool seeps. We've seen quite a few scapes in this area so far. And we saw a crab carrying an anemone. The latest fashion. Another skate. Yeah, sure. But I think we can continue searching around the basin. Around the pool. There's another one. Wow, that's a lot of skates. Yeah. Oh. You want to move into the bowl more? Yeah? Down and in. Yeah, so we kind of want to move down and into the bubbles. That way? Does that make sense? What I'm saying?
To the left.
To the left?
They're centered at. You see that circle there?
Yeah.
This is where we start.
That's where the sink is?
Yeah.
Okay. Yeah, we do love bubbles here.
Circle rolling.
Yeah. Fish.
Big fish.
Yeah, you wanna zoom in?
Thank you.
Yeah, can we take a zoom in on the big fish?
That's a good idea.
Oh,
Gosh.
ghost shot, yeah.
Still wanna linger or you wanna stick around and some more
Okay,
footage?
zoom.
Yeah? Got a zoom? Can we get a zoom in, please? And then let me know when to take them off. Yeah. Can we cut the lasers? So graceful. So strange looking. People in the room are really enjoying the chimera. Oh, that's nice. Just spamming.
Oh, shrimpy.
You good? Move on?
That's
Good.
this.
Okay, we're good. Yeah, we can continue
The vessel
searching.
30 meters to north.
I'm moving your desk so you can go there over there. Because there's another marker here in this area. According to the multi-beam, there's some bubbles. So we have more seepage on this area.
Turn stop. Yeah. Down the hill. So like down here, yeah? Yeah.
Just trying to understand more. Sorry.
You were asking something?
And that map, I don't know which direction I'm moving there.
All right. I mean,
Well,
You
well,
can make another pattern, but the one you will be making is maybe a bigger one.
bigger ones, no problems. We're just gonna go
It's like-
across that
it's
line
like-
in and out.
Yeah, it's like you don't want to go really far away from where you come to, because I really want to film where we were before. So we're just like exploring to have a better understanding of the area, and then we will come back to like where the bubbles were.
Can we do like grid lines? Because
Yeah,
if I could do
amazing.
like grids, I can
Okay,
follow the
yeah, that would
pattern
be
here.
great.
It'd be
Yeah,
better,
that would be great.
right? So I'm gonna start here.
great suggestion.
And then
another skate. Yeah, right? I don't know. So if anyone is just joining, we found a coal seep, a small coal seep. So we are kind of sticking near the area to see and exploring around to see if we see any other small coal seeps around. We're looking for some bubbles. And also just stopping to see if there's anything interesting nearby. So we were making some circles around the small seep, but now we're just gonna try some grid lines. Some relatively straight lines to explore. Seeing some large patches of sargassum. That's odd. Yeah, what are those? Speed.
I love this skate.
Do you want to zoom in on everyone?
Sorry?
No.
You don't want to zoom in on every sweep, right?
No.
There's two in me.
There's too many. Oh, it's two.
fun.
Interesting one.
Listen,
Very interesting one.
saw that.
Mm.
My color.
Great. Yeah, the sargassum one is like slowly rolling by on the seafloor. Looks like some tumbleweed.
Well, top left.
Top left? Oh, yes. Can you see that? That's big. It's a crab. You gonna stop and get some zoom in, or? Yeah, can we get stop and just zoom in for some pictures? With and without the lasers, please.
¿Qué es eso?
That's a big one. I think I get an enemy. Yes, please. Yeah, do we want to get in front? Can we take the lasers off for a minute? Maybe you want to put them back? Yeah, it's like a monster. It's a horror monster. It is so massive compared to so many other small things. Just walking through, picking up food. Really, it's unfunny.
That's true.
Good for you? Do you want to take from your front?
Where's the grass?
30 seconds.
Good to move on? Good to move on? Okay, I think we can go ahead. It's nice, huh?
Can I move? Or are you recording?
See, there's some kelp. Later.
Hmm?